Welcome back. Tonight we're turning our attention to something you've never seen, something so small it exists entirely beyond the reach of your naked eye, yet so abundant that it shapes every breath you take, every ecosystem on Earth, and perhaps even your thoughts as you drift towards sleep. We're exploring microorganisms, the invisible architects of reality itself. If you enjoy these gentle journeys through science, consider subscribing to the channel. It helps us continue creating these quiet explorations for nights just like this one. Now, let's begin. Microorganisms have existed for at least 3.5 billion years, making them Earth's oldest and most enduring life forms. Think about that for a moment. 3.5 billion years. Our species, Homo sapiens, has been around for perhaps 300,000 years. A blink in geological time. Dinosaurs roamed for about 165 million years, which feels impossibly ancient to us. But microorganisms, they were here first, shaping this planet for nearly 3 billion years before the first animals ever appeared. When Earth was young and violent, when asteroids still pummeled the surface, and the atmosphere bore no resemblance to the air you're breathing now, microorganisms were already here, already thriving, already beginning the slow work of transformation. They witnessed, or perhaps more accurately they were, the beginning of life's story on this planet. Everything else, all the complexity you see around you, all the forests and whales and hummingbirds and people, we're all late arrivals to a party that started billions of years ago. The microorganisms were here first, patient and persistent, were invisible pioneers that never left. A single teaspoon of soil contains more microorganisms than there are people on Earth. Picture it. That small amount of dirt you could balance on a spoon, perhaps from your garden or a potted plant, hosts more individual living things than the 8 billion humans currently alive. It's a difficult number to truly comprehend, isn't it? Each grain of soil is essentially a metropolis, a thriving city of microscopic life forms going about their business, eating, reproducing, competing, cooperating, dying, being reborn. You've walked across these invisible civilizations your entire life without knowing it. Every step you take presses down on billions of organisms living their complete life cycles in the darkness of the Earth. They don't know you exist, and until relatively recently in human history, you didn't know they existed either. Yet there they are, turning over nutrients, breaking down dead matter, making the soil fertile enough to grow the food that keeps you alive. That pinch of dirt in your garden isn't empty space between rocks. It's as densely populated as any city you've ever visited, just at a scale your eyes will never perceive. The word microbe comes from the Greek words mikros, meaning small, and bios, meaning life. Literally, tiny life. It's a simple, almost poetic name, though it wasn't coined until 1878 by a French surgeon named Charles Sédillot. Think about what that means. For all of human history before that moment, all the thousands of years of civilization, all the philosophy and art and science, we had no name for these organisms. We'd been dying from them, fermenting with them. It was depending on them utterly, yet we had no word to describe what they were. People had been making bread and beer and cheese for 10,000 years, harnessing these invisible workers without knowing they existed. They had been falling ill from infections, watching plague sweep through populations, never understanding the invisible agents behind the suffering. And then, in the late 1800s, we finally found the language to name them. Tiny life. Microbes. It's remarkable how long we lived in ignorance of our most ancient companions, and how recently we finally learned to see them, to name them, to begin understanding our place in their world. Antony van Leeuwenhoek first observed microorganisms in 1676 using a handmade microscope with only one lens. He wasn't a trained scientist, he was a Dutch draper, a fabric merchant with an unusual hobby, grinding lenses. The microscopes he created were astonishingly simple by modern standards, just single, carefully ground pieces of glass, yet they could magnify objects up to 270 times, and when Leeuwenhoek turned one of these lenses toward a drop of water, he saw something no human had ever witnessed before. He called them animalcules, little animals. In his letters to the Royal Society of London, written in Dutch and filled with wonder, he described these tiny creatures dancing and tumbling in drops of lake water, in rainwater, in scrapings from his own teeth. Imagine being the first person to see this hidden world, to realize that what appeared empty was actually teeming with life. He saw them moving, he saw their shapes, and he understood, perhaps dimly, that he had opened a door to something vast and previously inconceivable. The invisible world had become visible and nothing would be the same. Your body contains roughly the same number of microbial cells as human cells, about 37 trillion of each. You are quite literally as much microbial as you are human. This isn't an invasion or an infection. This is the normal state of being. You're not an individual organism so much as you are a walking ecosystem, a collaborative venture between your human cells and the trillions of bacteria, fungi, and other microorganisms that call your body home. They live on your skin, in your mouth, throughout your digestive system, in places you've never thought about. Some of them have been with you since the day you were born. Others joined later, acquired from the food you ate, the people you kissed, the soil you touched as a child. Together you form something larger than either component alone, a superorganism, a partnership so intimate that the boundaries between you and them become philosophical rather than biological. When you say I, you're speaking for trillions. You are never alone, not even in your most private moments. You are a universe of collaboration, a democracy of cells, human and microbial, working together in ways we're only beginning to understand. Microorganisms include bacteria, archaea, fungi, protozoa, algae, and viruses, though viruses are technically not alive, which is a strange thing to say about something that so clearly does things, that replicates and evolves and causes all manner of effects in the world. But by our strict definition of life, organisms that can reproduce independently, that maintain homeostasis, that metabolize, viruses fall just outside the boundary. They need a host cell to reproduce. They're more like sophisticated molecular machines than living things, though the line blurs in ways that make biologists uncomfortable. This diversity in the microbial world is staggering. When we group bacteria, archaea, fungi, protozoa, and algae, all under the umbrella term microorganisms, we're actually describing more variation, more fundamental difference, than exists between all plants and all animals combined. The differences between an oak tree and an elephant are, in evolutionary terms, relatively minor compared to the differences between a bacterium and an archaeon. The microbial world contains multitudes, different kingdoms of life, different strategies for survival, different biochemistries, all existing at scales too small for us to see without instruments. The invisible world is far more diverse than the visible one. The smallest known bacteria, Mycoplasma genitalium, measures just 200 to 300 nanometers across. To put that in perspective, you could line up 25,000 of them across the width of a single human hair. Think about that. A hair from your head, something you can barely see, could serve as a runway for thousands of these cells marching in formation, and you still wouldn't be able to see them even if you were looking directly at them. These organisms exist at a scale where the normal rules of our world start to feel strange. At that size, water doesn't flow like water anymore. Its viscosity becomes overwhelming, like swimming through honey. As Brownian motion, random jostling from surrounding molecules, becomes a significant force. These bacteria don't really swim the way we think of swimming. They're buffeted and pushed by thermal vibrations, navigating a molecular storm we can't even perceive. They've miniaturized life to its practical limits, pared down to just the essential machinery needed to survive and reproduce, tiny beyond comprehension, yet still undeniably alive. Bacteria come in three basic shapes: spheres, which we call cocci, rods, called bacilli, and spirals known as spirilla. It's a surprisingly simple geometry for such diverse organisms, three basic forms, yet within these simple shapes lies infinite variation in how they live, how they move, how they transform their worlds and survive in environments that would kill anything else. Here some spherical bacteria cluster together in chains like beads on a string. Some rods are short and stubby, others long and thread-like. Some spirals are loose and lazy, others tightly wound like corkscrews. The shape matters. It affects how they move through liquids, how they attach to surfaces, how they interact with their environment. But the shape is just the beginning. What matters more is what they do with that shape, the biochemical wizardry they perform inside their tiny cellular boundaries, the ways they've adapted these simple geometries to thrive in boiling acid, in frozen wastelands, in darkness, miles beneath the ocean, in your mouth, in the clouds, everywhere. Three shapes, infinite possibility. A single bacterium can divide into two cells in as little as 20 minutes under ideal conditions. One becomes two. 20 minutes later, two become 4, then 8, then 16. The mathematics are exponential, which means the numbers become staggering very quickly. One cell could theoretically become 500,000 cells in just 6 hours if conditions remained perfect, if resources never ran out, if nothing went wrong. Of course, conditions are never perfect for long. Resources always become limiting, space runs out, waste products accumulate, predators arrive. The exponential dream always hits constraints. But that potential, that capacity for explosive growth is always there, waiting. It's why bacteria can colonize new environments so quickly, why they can evolve so rapidly, why they're so successful. They reproduce faster than our immune systems can often respond, faster than antibiotics can sometimes kill them. Speed is their strategy, numbers their shield. They play the odds through sheer multiplication, betting that some descendants will survive even when most perish. Microorganisms can survive in boiling hot springs, in frozen Antarctic ice, miles underground in solid rock, and even in the water-cooling nuclear reactor cores. They've mastered survival in places we once thought were completely sterile, utterly hostile to life. Every time we've declared somewhere too extreme, we've later found microorganisms thriving there, indifferent to our assumptions about what life requires. They've expanded our definition of habitable beyond recognition. We used to think life needed moderate temperatures, liquid water, nutrients, sunlight, a narrow band of conditions we found comfortable. But microorganisms have shown us otherwise. Some prefer boiling acid to neutral water. Some eat rock instead of organic matter. Some thrive in pressures that would crush submarines, in radiation that would kill us in minutes, in cold that would freeze our cells solid. They've colonized every niche Earth offers, no matter how extreme, teaching us that life is far more tenacious, far more adaptable, far more stubborn than we ever imagined. The limits we perceived were not life's limits. They were merely the limits of our imagination. The first microorganisms arose in Earth's oceans when our planet was still being bombarded by asteroids, when the sky was perpetually clouded with dust and vapour, when the surface was anything but peaceful. Life didn't begin in tranquility. It began in chaos, in violence, amid impacts that would have shattered any modern organism instantly. Yet somehow, in that hostile environment, chemistry became biology. Molecules organized themselves into patterns that could persist, that could replicate, that could adapt. It's humbling in a way. We think of life as fragile, something that needs protection, careful conditions, stability. But the first life emerged during what we'd consider an apocalypse. Perhaps there's something profound in that. Life didn't wait for perfect conditions. It found a way forward in the midst of catastrophe, clinging to existence in scalding vents on the ocean floor, in pools of water briefly sheltered from the bombardment, anywhere chemistry could build upon itself. Those first microorganisms, whose descendants still live in you and around you, were forged in chaos. They didn't need peace. They needed only possibility, and they found it in Earth's most violent epoch. Early microorganisms called cyanobacteria invented photosynthesis around 2.4 billion years ago. Invented, that's the right word, isn't it? They developed a completely new way of harvesting energy, a biochemical innovation that would reshape the entire planet. Before cyanobacteria, Earth's atmosphere contained almost no oxygen. The air would have suffocated you instantly. But these ancient bacteria learned to split water molecules using sunlight, releasing oxygen as a waste product. This single innovation, this molecular machinery that captured light and turned it into chemical energy, made everything else possible. Every breath you take, every complex organism that's ever lived, owes its existence to cyanobacteria's invention 2.4 billion years ago. Trees didn't invent photosynthesis. Plants didn't invent it. Microorganisms did. And they did it when Earth was still young, when the continents were just beginning to form, when the only life anywhere was microscopic. They changed the air. They changed the chemistry of the oceans. They created the conditions that would eventually allow creatures like you to exist, to breathe, to think about the ancient bacteria that made it all possible. The great oxidation event caused by photosynthetic microbes was actually toxic to most life at the time. Oxygen, the molecule you depend on for every moment of consciousness, was poison to early organisms. It's a reactive molecule, corrosive, dangerous to cells that hadn't evolved defenses against it. When cyanobacteria started pumping oxygen into the atmosphere as waste, they were essentially polluting the environment for everyone else. Yet it must have been catastrophic for anaerobic organisms, those that thrived in the oxygen-free world that had existed for over 1 billion years. They retreated, forced into oxygen-free refuges in mud and deep water and intestines, anywhere the corrosive gas couldn't reach. Meanwhile, new organisms evolved that could tolerate oxygen, then use it, then depend on it. What was one organism's waste became another's necessity. It's the ultimate example of how life recycles, how catastrophe for some becomes opportunity for others, how the composition of air itself is negotiable, changeable, determined by whoever happens to be doing the breathing. We live in an oxygen world now, but it wasn't always so, and it required a mass poisoning to get here. Stromatolites, those strange rock-like structures built by layered microorganisms, are some of Earth's oldest fossils, preserved evidence of ancient microbial communities that date back over 3 billion years. They're not quite rocks, and not quite alive, but something in between. Living communities that slowly build mineral structures layer by layer, year after year, creating formations that can persist long after the organisms themselves have died. Living stromatolites still grow in a few places today, in Shark Bay in Australia, in some hypersaline lakes, places where conditions happen to favour their slow, patient construction. They're architectural echoes of the ancient world, essentially unchanged in billions of years when you look at a stromatolite. You're seeing a structure being built the same way it was built before animals existed, before plants, before fungi, when microorganisms were the only show on Earth. They're time capsules, living fossils, bridges between the deep past and the present. The same processes that built rocks 3 billion years ago are still building them today, indifferent to the vast changes that have transformed everything else. Mitochondria, those organelles inside your cells that produce energy, were once free-living bacteria that got absorbed billions of years ago and never left. Every breath you take is powered by the descendants of ancient bacterial immigrants. It's called endosymbiosis, one organism living inside another in a mutually beneficial relationship so intimate that they became inseparable. Imagine it. Billions of years ago, some larger cell engulfed a bacterium, perhaps intending to digest it, but the bacterium survived inside, protected and fed, and in return it generated energy more efficiently than the host cell could manage alone. Both prospered. Neither could afford to leave. Over time, the bacterium transferred most of its genes to the host cell's nucleus, streamlined itself, became specialized. It stopped being an independent organism and became an organelle, a part rather than a whole. But it retained just enough of its own DNA to remind us of its origin. Your mitochondria are domesticated bacteria, ancient immigrants that have lived inside eukaryotic cells for so long that neither can survive without the other. You carry their descendants in every cell, billions of them, working ceaselessly to keep you alive. Some microorganisms can exchange genetic material horizontally, swapping DNA like trading cards, sharing genes without needing to reproduce. It's called horizontal gene transfer, and it's utterly unlike how we inherit traits. You get your genes from your parents, a vertical transfer through generations. But bacteria can acquire genes from neighbours, from viruses, from dead cells decomposing nearby, incorporating useful DNA into their own genomes, sometimes in a matter of hours. This means they don't need to wait for reproduction to evolve. If one bacterium develops antibiotic resistance, it can share that gene with others around it, even bacteria from different species. Useful adaptations spread through populations, not just across generations, but across individuals, sideways through communities, creating genetic fluidity we're only beginning to understand. It's evolution on fast forward, adaptation at a pace that sometimes outruns our ability to respond. They trade survival strategies like currency, borrowing innovations from each other in real time, solving problems collectively rather than individually. It makes them extraordinarily adaptable, frustratingly difficult to eliminate, and endlessly inventive. Endospores are dormant bacterial structures that can survive for millions of years in suspended animation, waiting patiently for conditions to improve. When a bacterium faces starvation, desiccation, or other lethal stresses, some species can create these incredibly durable packages, protective shells containing their genetic material and just enough cellular machinery to restart life when circumstances change. We scientists have revived bacteria from 250 million-year-old salt crystals, awakening organisms that had been dormant since the Triassic period, before dinosaurs dominated the Earth, before birds existed, before flowering plants. Think about that. A bacterium went dormant when the continents were still joined in Pangaea, and it waited metabolically inert, essentially dead but not quite, for 250 million years, and then, given moisture and nutrients, it simply resumed living, as if no time had passed at all. These endospores can survive boiling, freezing, radiation, chemicals that would dissolve normal cells. They're among the most durable biological structures known, tiny time capsules waiting for better days. Perhaps some are waiting still, in ancient rocks, in permafrost, in sealed caves, yet carrying genetic memories of worlds long vanished. Archaea, discovered in 1977, are so fundamentally different from bacteria that they constitute their own domain of life entirely. For over a century, we had grouped them with bacteria, completely missing the distinction, lumping together two fundamentally different forms of life because they looked similar under microscopes. It took sophisticated molecular analysis to reveal the truth. Archaea are as different from bacteria as we are from bacteria. Carl Woese made the discovery by comparing ribosomal RNA sequences, essentially reading the molecular signatures of these organisms, and realizing that some of what we'd been calling bacteria were actually something else entirely, something ancient and strange. He proposed a new tree of life with three main branches: bacteria, archaea, and eukarya, which includes us, instead of the two we'd assumed. It was revolutionary and controversial, a fundamental restructuring of how we understood life's diversity. Many archaea live in extreme environments: boiling springs, salt lakes, deep-sea vents, acidic pools, places we once thought only bacteria could tolerate. But they're not bacteria. They're their own thing, using different molecular machinery, different metabolic strategies, different cellular chemistry. We had been blind to them for so long, seeing but not recognizing, another reminder of how much remains hidden even in what we think we understand. Microorganisms evolved every major biochemical process that life uses: photosynthesis, respiration, fermentation, nitrogen fixation, all of it. Long before plants, long before animals, even microorganisms had already invented the chemistry that powers ecosystems. They perfected these processes over billions of years, refined them, optimized them, created the metabolic diversity that makes the living world possible. When you breathe, you're using respiration, a process bacteria invented. When plants photosynthesize, they're using machinery originally developed by cyanobacteria. When your muscles ferment glucose during intense exercise, that's another microbial innovation. The chemistry that keeps us alive, that keeps all complex life functioning, isn't ours, it's borrowed. We inherited it from microorganisms, incorporated it into our cells, depend on it absolutely, but we didn't invent it. all the chemistry that matters, all the molecular transformations that capture energy and build structures and cycle nutrients through ecosystems, was worked out by invisible pioneers billions of years before we arrived. We are latecomers to chemical pathways already ancient beyond measure. The recipes that run in our cells were written by microorganisms in Earth's first eons, and we're still following their instructions. Some bacteria can survive 5,000 times the radiation dose that would kill a human, repairing their shattered DNA with an efficiency that seems almost miraculous. Deinococcus radiodurans, nicknamed Conan the Bacterium, can endure radiation levels found in nuclear reactor cores, in radioactive waste, in the vacuum of space. Doses that would tear your DNA into hundreds of fragments, causing rapid death, barely slow these bacteria down. How do they do it? They maintain multiple copies of their genome, so when radiation shatters the DNA, they have backup templates. They possess extraordinarily efficient DNA repair enzymes that can reassemble their genome from hundreds of fragments, like solving a complex molecular jigsaw puzzle. They can rebuild their genetic code from scratch, essentially reconstituting themselves after being blown apart at the molecular level. It challenges our assumptions about biological limits, about what's possible for life. These bacteria aren't particularly exotic in most ways. They live in soil, in water, in ordinary environments. They don't need radiation to survive, but they can tolerate it, endure it, repair the damage it causes with a proficiency that makes our cellular repair mechanisms look primitive by comparison. Perhaps they evolved this ability to survive desiccation, which also fragments DNA, and radiation resistance came along as a side effect. Whatever the reason, they've mastered survival in conditions that seem incompatible with maintaining genetic integrity, and they do it quietly, without fanfare. Just another invisible resilience we've discovered in the microbial world. Bacteria can communicate with each other through chemical signals in a process called quorum sensing, essentially counting their neighbours and coordinating group behaviours based on population density. They release signalling molecules that accumulate in the environment, and when the concentration reaches a certain threshold, indicating enough bacteria are present, they collectively change their behaviour. It's a form of microbial democracy, in invisible elections held in chemical gradients. When bacteria are few and scattered, they act as individuals, but when their numbers grow, when the quorum is reached, they switch strategies. They might start producing light together, creating bioluminescence bright enough to see. They might form biofilms, building protective communities. They might release toxins, coordinate attacks, express genes that only make sense in groups. Individual bacteria are blind to each other in the traditional sense, but through chemistry they can sense the presence of their kin, gauge their numbers, and act collectively. It raises strange questions. How does a single bacterium decide to participate in group behaviour? What drives coordination among cells with no central command, no leaders, no consciousness we'd recognise? Yet somehow they manage it, forming microbial communities with division of labour, with cooperation, with collective responses that emerge from countless individual chemical conversations. They're not thinking, not planning, yet the outcome resembles coordination, resembles decision-making, patterns of collective behaviour arising from simple molecular rules repeated billions of times. Some bacteria produce magnetite crystals inside their bodies and use Earth's magnetic field to navigate, orienting themselves in the darkness like living compasses. These magnetotactic bacteria create chains of tiny magnetic particles, each crystal perfectly formed, aligned in rows that give the cell a north-south polarity. They don't think about magnetic fields the way we do. They simply follow the field lines, letting Earth's magnetism guide them through sediments and water columns. Or in the northern hemisphere, they typically swim toward magnetic north, which angles downward, pulling them toward deeper oxygen-poor waters where many species prefer to live. In the southern hemisphere, the polarity reverses. Bacteria there follow south, which points down in that part of the world. It's an elegant navigation system, utterly passive, requiring no energy, no computation, just iron crystals arranged in chains, and the planet's magnetic field gently tugging them toward favourable environments. Imagine being so small that you navigate by magnetism, that the invisible field permeating the planet becomes your primary sense of direction. They're swimming through a world we can only detect with instruments, responding to forces we can't feel, using senses we don't possess. Every environment contains invisible gradients these bacteria perceive: magnetic, chemical, thermal, a sensory landscape we walk through blindly while they navigate it with precision. Bioluminescent bacteria create the ethereal glow seen in disturbed ocean water at night. Those sparkles and flashes when you drag your hand through dark waves, or when boats leave glowing wakes behind them. Each flash is millions of bacteria producing cold light through chemical reactions, coordinating through quorum sensing so they only glow when their numbers are high enough to make a visible display. The light is created when luciferin molecules react with oxygen in the presence of the enzyme luciferase, releasing energy as photons instead of heat. It's a clean reaction, almost perfectly efficient, no warmth wasted, just pure luminescence generated through biochemistry. The bacteria often live symbiotically inside marine animals, squid, fish, jellyfish, providing light in exchange for shelter and nutrients. Some fish use bacterial light organs as lures to attract prey in the deep sea. Some squid use them as camouflage, matching the downward glow of sunlight filtered through water above them. But sometimes the bacteria simply bloom free in the water, and when conditions disturb them, a wave breaking, a swimmer moving through the darkness, they flash in response, turning the ocean into liquid stars. It's one of nature's quieter magics. Bioluminescence created not by special organs evolved over millions of years, but by single-celled organisms doing chemistry in the dark, transforming the night sea into something dreamlike and strange. Bacteria in your mouth form complex architectural structures called biofilms, what you know as dental plaque. They're not just random accumulations of cells, they're organized communities with structure, with architecture, with channels for water flow and waste removal. Different bacterial species occupy different layers, creating vertical stratification. Some bacteria anchor themselves to your tooth enamel, forming the foundation. Others build on top of them, creating towers and mushroom-shaped structures. Some create water channels that flow through the biofilm like streets through a city. There's even division of labour. Some bacteria produce sticky polymers that hold the community together. Others break down nutrients. Some protect the colony from your immune system or from antibiotics. The biofilm functions as a unit, just resistant to forces that would kill individual bacteria. Antibiotics that can kill bacteria swimming freely in liquid often can't penetrate the biofilm's protective matrix. Your immune cells can't easily reach bacteria deep inside these structures. It's a microbial city built on your teeth, constructed from cells and polymers, complete with infrastructure and specialized roles. Every time you brush your teeth, you're essentially demolishing these bacterial settlements, scraping away their architecture, but they simply rebuild once you stop, patient and persistent, reconstructing their communities from whatever cells survive. Some bacteria can glide across surfaces without flagella, those whip-like tails many bacteria use to swim. And scientists still don't fully understand the mechanisms involved. They seem to flow like liquid, be moving smoothly across solid substrates in ways that don't match our understanding of how cells should propel themselves. Some appear to secrete slime that they push against. Others might use tiny protein motors distributed across their surface. Still others might employ mechanisms we haven't even identified yet. Watching them move is mesmerizing in time-lapse microscopy. They slide along, sometimes alone, sometimes in coordinated groups, leaving trails behind them, migrating across surfaces with purposeful directionality, but without obvious means of propulsion. No visible structures pushing or pulling them, no cilia beating, no flagella rotating, just smooth, continuous motion that defies easy explanation. It's humbling to realize that something as seemingly simple as bacterial movement can still contain mysteries, that even after centuries of microscopy and molecular biology, we can watch bacteria do things we can't fully explain. They've evolved solutions to the problem of movement that we're still trying to understand, using physics at the microscale in ways that don't scale up to our world, operating under rules that become clear only when you're small enough, when surface forces dominate over gravity, when viscosity becomes the primary challenge. The bacterium Thiomargarita namibiensis is visible to the naked eye, as large as the period at the end of this sentence, sometimes even bigger, up to 0.75 millimeters across. It challenges the very definition of microorganism, a giant among the typically invisible. When scientists first found these bacteria in ocean sediments off the coast of Namibia, they could actually see them as tiny white dots without any magnification at all. How did it get so large? Most bacteria remain small because they rely on diffusion to move nutrients in and waste out, and diffusion only works efficiently over short distances. But Thiomargarita solved this problem by becoming mostly empty space. The cell is filled with a giant vacuole, essentially a water-filled bubble, containing nitrate it uses for energy. The actual living part of the cell is compressed into a thin layer around the outside. Keeping the distance, substances must diffuse reasonably short despite the cell's overall size. It's an elegant solution to a fundamental constraint. By becoming hollow, it can be huge while still functioning like a bacterium. It's still a single cell, still governed by the same basic rules as its microscopic relatives, just scaled up in a way that makes it perceptible. A bacterium that crossed the visibility threshold and became something you could almost see, a bridge between the microscopic and macroscopic worlds. Certain bacteria can generate electrical currents and form living wires that conduct electricity across distances, creating microscopic power grids in mud and sediments. They're called cable bacteria, and they essentially function like biological electrical cables, transferring electrons from one environment to another, connecting oxygen-rich zones near the sediment surface with oxygen-poor zones deeper down. They form filaments, chains of cells connected end to end, sometimes stretching several centimeters long, which is enormous at bacterial scale. Electrons flow through these filaments, creating measurable electrical currents in the mud, since they're mining energy from chemistry in ways we're only beginning to understand, using electron transport over distances that seem impossible for organisms without specialized conductive tissues. Some bacteria in deep-sea environments appear to eat electrons directly from minerals, essentially breathing rocks instead of oxygen. Others dump electrons into minerals using solid metals as electron acceptors the way we use oxygen. They've created metabolisms based on electrical gradients, on the flow of charge through their environment. It's a completely different strategy for harvesting energy, one that doesn't require oxygen or light or organic molecules, just chemistry, conductivity, and the patient flow of electrons through mud in the darkness. Some microorganisms enter a zombie-like state called VBNC, viable but non-culturable, where they're alive but won't grow, existing in a twilight zone between life and death. They maintain intact membranes, they keep their DNA, they remain metabolically active at very low levels, but they refuse to divide when placed in culture media. They won't form colonies on petri dishes. By traditional microbiological methods, they appear dead or absent, yet they persist. Under the right conditions, often unknown to us, they can resuscitate, suddenly returning to normal growth and reproduction. It's as if they're waiting, dormant but aware, monitoring their environment for signals we don't understand, remaining in stasis until something tells them it's safe or advantageous to wake up. This has profound implications. When we test water for pathogens and find nothing growing in our cultures, are we detecting absence or merely overlooking dormant cells waiting to revive? They exist in a state we struggle to categorize. Not dead because they can return. Not alive in the sense of growing and reproducing. Somewhere in between, in a condition that suggests life has more modes than we typically acknowledge, that the binary of alive or dead doesn't capture the full range of possibilities available to microorganisms who've been perfecting survival strategies for billions of years. Fungi can form networks spanning acres underground, sharing nutrients between trees in forest internet systems called mycorrhizal networks. What looks like separate mushrooms above ground, fruiting bodies popping up across the forest floor, may actually be one enormous organism below, a continuous web of thread-like hyphae connecting tree roots across vast distances, facilitating communication and resource exchange between plants that seem separate but are actually linked. Trees in a forest aren't isolated individuals, they're nodes in a network, connected by fungal mycelium that transfers nutrients, water, even chemical signals between them. A large tree with deep roots and good access to water might share with younger trees in dry shade. Trees under pest attack might send warning signals through the network, allowing neighbours to activate defences before the pests arrive. The fungi facilitate these exchanges, taking a small percentage of the sugars the trees produce through photosynthesis in return for vastly expanding the tree's access to soil nutrients and water. Some of these fungal networks are ancient, older than the oldest trees they connect. They persist as trees die and new ones grow, as forests cycle through generations, the underground web remaining continuous, a living infrastructure that predates and outlives its individual members. The forest is more connected than it appears, more collaborative, unified by invisible threads carrying resources and information in the darkness beneath our feet. Certain bacteria can transform toxic mercury into harmless forms, performing environmental cleanup at the molecular level, essentially detoxifying poison cell by cell. They evolved these abilities to survive in mercury-contaminated environments, developing enzymes that chemically modify mercury into less toxic compounds or into gaseous forms that escape into the atmosphere. It's a defense mechanism, a way to survive in places that would kill other organisms, but it accidentally makes them nature's decontamination specialists. We're studying these bacteria, trying to understand and harness their abilities for bioremediation, using living organisms to clean up pollution. Imagine contaminated sites slowly healing themselves through bacterial action, mercury gradually converted and removed through biological processes rather than expensive chemical treatments. The bacteria don't know they're cleaning up our mess. They're just surviving, doing what they've evolved to do, and in the process they might help repair damage we've caused. It's a reminder that evolution solves problems we didn't know needed solving. These bacteria developed mercury resistance long before humans started mining and releasing mercury into the environment. They were ready, equipped with molecular machinery that just happened to be useful for addressing a problem we created. Perhaps the solutions to our environmental challenges already exist somewhere in the microbial world, waiting to be discovered in organisms that evolved different chemistries for their own purposes. Microorganisms in the ocean produce more than half of Earth's oxygen, more than all forests combined. When you think of oxygen production, you probably picture rainforests, towering trees with vast canopies converting carbon dioxide into the air you breathe. And trees do contribute, certainly. But the invisible organisms drifting in the ocean's sunlit surface layers, phytoplankton, cyanobacteria, microscopic algae, they're the true oxygen factories of our planet. Every other breath you take comes from the ocean, from photosynthetic microorganisms you'll never see, countless trillions of them floating in currents, capturing sunlight, splitting water molecules, releasing oxygen as waste. They're so small that thousands could fit on the head of a pin, yet collectively they outproduce every forest, every grassland, every terrestrial ecosystem combined. The ocean appears empty from above, just blue water extending to the horizon, but those upper sunlit layers are teeming with microscopic life performing the chemistry that keeps the atmosphere breathable. If these organisms faltered, if ocean conditions changed enough to reduce their population significantly, we'd notice in the composition of air itself. The oxygen you're breathing right now, some of it was produced moments ago by a microscopic cell floating somewhere in the Pacific, or the Atlantic, or the Indian Ocean, an organism that will never know you exist but contributed to this breath, and the next, and the one after that. You're connected to them, dependent on them, breathing the waste products of invisible ocean drifters who've been oxygenating the planet for billions of years. Nitrogen-fixing bacteria in soil convert atmospheric nitrogen into forms plants can use, making agriculture, and most terrestrial life, possible. The air around you is 78% nitrogen, an abundance of this essential element floating everywhere, yet most organisms cannot access it. Nitrogen gas in the atmosphere is chemically stable, locked in triple bonds that are incredibly difficult to break. Plants cannot use it. Animals cannot use it. We breathe it in and breathe it out unchanged, surrounded by nitrogen we cannot touch. But certain bacteria possess the enzyme nitrogenase, which can break those triple bonds and convert atmospheric nitrogen into ammonia, a form plants can absorb through their roots. These bacteria often live in nodules on the roots of legumes, beans, peas, clover in a symbiotic partnership. The plant provides sugars, the bacteria provide accessible nitrogen. Without them, most plants would starve in an atmosphere that's mostly nitrogen they cannot reach. Before humans learned to synthesize nitrogen fertilizers industrially, a process that requires immense heat and pressure. All the nitrogen in ecosystems, in crops, in your body's proteins and DNA came through these bacteria. They were the bottleneck, the essential step that made nitrogen available to the living world. Even now, with industrial fertilizer production, natural nitrogen fixation by bacteria still contributes enormously to global nitrogen cycles. They're still performing this essential service, still cracking atmospheric nitrogen and making it accessible, still enabling life to build proteins and nucleic acids from air that would otherwise be unusable. A single cow's digestive system contains trillions of microorganisms that break down cellulose from grass, performing digestion that the cow itself cannot manage. The cow is merely the vessel, the walking fermentation chamber. It's the microbes doing the actual work, the ones dismantling the tough plant fibers, extracting energy and nutrients from cellulose that would otherwise pass through undigested. Cows, like most animals, don't produce cellulase, the enzyme needed to break down cellulose. They can't digest grass on their own, but their specialized stomachs provide perfect conditions for microorganisms that can. The rumen, the largest chamber of a cow's stomach, is essentially a microbial bioreactor, maintained at ideal temperature and pH, constantly supplied with chewed plant material. Bacteria, protozoa, and fungi work there in vast numbers, breaking down cellulose into simpler compounds the cow can then absorb. It's a partnership, an ancient symbiosis. The cow provides shelter and steady food supply. The microorganisms provide digestive capabilities the cow lacks. Neither can thrive without the other. When you think about it, herbivores feeding on grass aren't really herbivores at all. They're microbial cultivators, farming bacterial populations in their guts, harvesting the products of microbial metabolism. The cow eats grass, but it lives on what the microbes make of it. Microorganisms decompose dead matter, recycling nutrients that would otherwise remain locked in corpses forever, unavailable to the living. Death feeds life, but only because of invisible undertakers dismantling the dead molecule by molecule, liberating carbon and nitrogen and phosphorus, returning elements to the soil and water and air where they can be used again. Without decomposers, dead things would accumulate. Their forests would pile up with fallen trees that never rotted. Bodies would mummify rather than returning to earth. Nutrients would be sequestered in an ever-growing mountain of the dead, gradually depleting the resources available to the living. But bacteria and fungi prevent this. They arrive at every death site, sometimes within minutes, and begin the work of disassembly. They produce enzymes that break down proteins, lipids, carbohydrates, even lignin and chitin and bone. They work in succession, different species specializing in different stages of decay, different compounds, creating an ecological progression from fresh death to complete decomposition. They transform bodies into soil, complexity into simplicity, organization into raw materials. It's not pleasant to contemplate, perhaps, but it's essential. The elements in your body have been recycled countless times, passed through innumerable organisms, decomposed and rebuilt again and again. You're made from recycled materials, atoms liberated from previous lives by microorganisms who never stop working, never stop dismantling, never stop feeding the living with the remains of the dead. Some bacteria can break down plastic, though far too slowly to solve our pollution crisis yet. Plastic was invented less than a century ago. Synthetic polymers that didn't exist in nature, yet already evolution is catching up, creating enzymes for materials that are newer than your grandparents. in landfills, in the ocean, in contaminated soil. Bacteria encounter plastic, and occasionally, rarely, one evolves the ability to digest it. The enzymes work slowly, inefficiently, because they weren't optimized for plastic. They evolved for other purposes and just happened to work on these synthetic polymers by accident. But they work. Scientists have found bacteria that can break down PET plastic, the kind used in water bottles, though it takes weeks or months to degrade what we discard in seconds. Other bacteria show promise with polyurethane, with polystyrene, with various plastics we assumed were essentially permanent. Evolution is patient. Given enough time, given enough bacteria encountering enough plastic, the mechanisms will improve. Mutations that make the enzymes slightly more efficient will spread. Bacteria will optimize their plastic digesting abilities the way they've optimized everything else over billions of years. Maybe in a few million years, plastic will be as biodegradable as wood. But we don't have millions of years. We need solutions now, and evolution's pace, though inexorable, is slower than our crisis. Still, it's happening. Life is adapting to our synthetic world, slowly learning to digest materials that didn't exist when we started polluting. Microorganisms in your gut produce vitamins K and B12 that your body cannot make on its own. You've outsourced these essential functions to residents who've lived in your intestines since birth. Microbial chemists brewing molecules you require but cannot synthesize. Vitamin K is crucial for blood clotting. Vitamin B12 is essential for nerve function and red blood cell production. Without them, you'd develop serious deficiencies, yet you don't manufacture either one yourself. Your gut bacteria do it for you, as part of their own metabolism. They're not doing it to help you, exactly. They're doing what they need to survive, and the vitamins are byproducts, excess molecules that leak out and happen to be exactly what you need. It's almost too convenient, too perfectly matched. But it makes sense when you remember how long this partnership has existed. Humans and gut bacteria have been co-evolving for millions of years, each shaping the other's evolution, creating dependencies and symbioses so deep that the line between individual and ecosystem blurs. You could get these vitamins from your diet, from fermented foods or animal products, but your gut bacteria provide a backup supply, a continuous low-level production that helps ensure you don't become deficient. They're working right now as you read this, as you drift towards sleep, producing molecules your cells need, contributing to your health in ways you never consciously consider. You're never truly alone, never independent. You're a collaborative venture, and some of your essential biochemistry happens not in your cells, but in theirs. Antarctic bacteria survive by producing antifreeze proteins that prevent ice crystals from forming in their cells, allowing them to remain liquid and alive at temperatures that would freeze and shred normal organisms. Ice is dangerous to cells because water expands when it freezes, forming crystals with sharp edges that can puncture membranes and destroy cellular structures. But these bacteria have evolved proteins that bind to tiny ice crystals as they form, preventing them from growing larger, essentially capping the crystal growth and keeping the interior of the cell from solidifying. They've mastered living in permanent winter, in temperatures that never rise above freezing, in environments where liquid water is rare and precious. Some live in Antarctic ice itself, in tiny liquid veins and pockets within glaciers, surviving in microscopic habitats carved in frozen landscapes. Others live in supercooled lakes that remain liquid below freezing point, stabilized by salt or pressure, or the antifreeze proteins the bacteria themselves produce. We're studying these proteins now, trying to understand and replicate them for frozen food storage, for organ preservation, for protecting crops from frost damage. The bacteria evolved these molecules for their own survival, but they might help us solve human problems, might protect tissue and food and plants from ice damage. It's another case of evolution producing solutions we can borrow, of organisms inventing technologies through natural selection that we can adapt for our purposes, molecular innovations developed in the harshest environments on Earth. Some microorganisms can digest petroleum and crude oil, breaking down ancient sunlight stored in carbon chains, dismantling molecules that formed from the compressed remains of organisms that died millions of years ago. These bacteria possess enzymes that can crack open hydrocarbons, liberating energy from oil by oxidizing it, essentially breathe in petroleum the way we breathe oxygen. They helped clean up oil spills long before humans knew they existed. When the Deepwater Horizon spilled millions of barrels of oil into the Gulf of Mexico, bacteria bloomed in response. Populations exploding as they fed on the sudden abundance of hydrocarbons. They consumed the oil, broke it down, transformed it into carbon dioxide and biomass, slowly processing the spill through countless cellular metabolisms. They couldn't work fast enough to prevent ecological damage, but they accelerated the cleanup, reduced the oil's persistence, invisible emergency responders working around the clock. These bacteria weren't created by the spill. They've always existed in ocean environments, in soil, anywhere hydrocarbons naturally seep from the Earth. Oil has been leaking from the planet's crust for hundreds of millions of years, creating natural seeps that release thousands of tons annually, and bacteria evolved to exploit this resource. They were ready when our tankers spilled, when our wells blew out. Equipped with enzymes refined over evolutionary time, they're still working wherever oil meets water, wherever hydrocarbons enter ecosystems, patient and persistent, eating pollution one molecule at a time. Microorganisms drive the entire carbon cycle, moving carbon between air, water, earth, and living things, mediating the transformation and transfer of this essential element through all of Earth's systems. They're not the only participants. Plants photosynthesize, animals respire, volcanoes emit. But microorganisms are involved in nearly every step, every transformation, every pathway carbon takes. Photosynthetic bacteria and algae fix carbon dioxide from the atmosphere, converting gas into organic matter. Decomposers break down dead organisms, releasing carbon back to the atmosphere or into soil. Bacteria in ocean sediments bury carbon in the deep sea, sequestering it for millions of years. Methanogens produce methane in wetlands and ocean sediments, releasing a potent greenhouse gas. Other bacteria oxidize methane, consuming it before it reaches the atmosphere. The carbon you exhale was probably processed by microorganisms multiple times before you incorporated it into your tissues. Climate itself is partially regulated by organisms too small to cast shadows. The balance of carbon dioxide in the atmosphere, the temperature of the planet, the habitability of Earth, all influenced by microbial activity occurring at scales we can barely perceive. When we talk about climate change, we're partly talking about disrupting microbial processes refined over billions of years. There's overwhelming natural carbon cycles with fossil carbon released too quickly for biological systems to absorb. The microorganisms will continue cycling carbon, as they always have, but we've altered the flows, changed the ratios, pushed the system into new territory where the old equilibriums no longer hold. Termites rely on gut microorganisms to digest wood. Without them, termites would starve on their wooden diet, unable to extract any nutrition from the cellulose they consume. A termite's gut is a carefully maintained microbial ecosystem, containing protozoa and bacteria that possess the cellulase enzymes the termite lacks. The termite chews wood into small particles, creating ideal conditions for microbial digestion, and the microorganisms break down cellulose into compounds the termite can absorb. What seems like a simple pest eating the framing of your house is actually a walking fermentation chamber, a collaboration between kingdoms. The termite is the farmer, cultivating microbial populations, maintaining perfect conditions in its gut for the bacteria and protozoa it depends on. The microorganisms are the labourers, doing the biochemical work of digestion. Neither can survive without the other. Young termites acquire their gut microbes by consuming the feces of older termites, inheriting not just genes from their parents, but also microbial partners essential for survival. This partnership is so ancient, so fundamental, that termites have evolved elaborate social behaviours to ensure every individual acquires the right microorganisms. The colony maintains and propagates not just termite genetics, but also microbial cultures, passing down invisible symbiotic partners generation after generation. When you see termite damage, you're seeing the result of this ancient collaboration, countless microorganisms dismantling cellulose inside countless termite guts, an alliance between visible and invisible life working together to transform wood into energy. Humans have been using yeast for baking bread and brewing alcohol for at least 10,000 years, harnessing invisible workers we didn't know existed, managing microbial processes we couldn't see or understand. Our civilizations were built partially on fermentation, on the biochemistry of single-celled fungi converting sugars into carbon dioxide and ethanol, leavening our bread and intoxicating our beverages. Ancient bakers didn't know about yeast. They just knew that if you mix flour and water and left it exposed to air, something happened. The dough would rise, it would develop flavour. Bread baked from this transformed dough was lighter, more digestible, more delicious than flatbread. So they saved portions of successful dough as starter cultures, propagating whatever had colonized it, passing these starters down through generations like family heirlooms. Some sourdough starters in use today are over a century old, continuous cultures maintained and fed weekly, the same microbial community sustained across decades. The same with brewing. Leave grape juice or grain mash exposed, and it ferments. Ancient people didn't understand why, but they perfected the conditions, the timing, the temperatures that produced the best results. Yeah, they were practicing applied microbiology thousands of years before microbiology existed as a science, domesticating microorganisms through trial and error, creating partnerships with yeast that shaped human culture. Beer and bread civilizations, built on invisible labour, on chemistry we harnessed before we knew it was chemistry at all. The distinctive flavours of cheese, yogurt, sourdough and kimchi all come from specific microbial communities, each culture producing its own chemical signature, its own blend of acids and alcohols and aromatic compounds. Cuisine is applied microbiology, though we rarely think of it that way. When you taste sharp cheddar or tangy yogurt or funky blue cheese, you're tasting the metabolic byproducts of bacteria and fungi, the molecules they produce while transforming milk or cabbage or dough. The different microbes create different flavours. The bacteria in yogurt produce lactic acid, creating that characteristic tang. The moulds in blue cheese produce compounds that taste pungent, almost overwhelming, polarizing. The mixed culture in sourdough, wild yeast and lactic acid bacteria working together, creates complex flavour profiles that vary by region, by the specific microbial communities present in each bakery's environment. Traditional kimchi involves multiple fermentation stages, different bacteria dominating at different times, each contributing different flavours and textures. These aren't random processes, they're carefully managed, though often through tradition rather than scientific understanding. Temperature, salt concentration, oxygen availability, all these variables select for certain microbes over others, shaping the community and therefore the flavour. A cheesemaker managing a culture is curating an ecosystem, fostering specific species and strains, creating conditions that produce desired outcomes. It's a craft of managing invisible cultures, of guiding fermentation through countless microbial generations, transforming bland ingredients into complex flavours through biological alchemy we've practiced for millennia. Your unique body odour is determined not by you, but by the specific microorganisms living on your skin, their metabolic activities producing the volatile compounds that become your scent. The sweat your body produces is actually nearly odourless when fresh. It's mostly water and salt. But the bacteria living on your skin metabolize compounds in that sweat, and breaking down proteins and lipids into smaller molecules that evaporate and reach other people's noses. Different bacterial species produce different smell compounds. The bacteria in your armpits produce different odours than those on your feet, than those on your scalp. The specific mixture of species colonizing your skin, shaped by your genetics, your hygiene habits, your environment, your diet, creates a scent signature as unique as your fingerprint. Identical twins, despite sharing DNA, develop different body odours because they cultivate different skin microbiomes. You smell like the microbial garden you cultivate, like the billions of residents metabolizing on your surface. When someone recognizes you by scent, when you smell different after changing deodorants or diets or antibiotics, that's your microbiome changing. Different species rising and falling in abundance, altering the chemical bouquet you emit. You're not just carrying these organisms, you're broadcasting their presence, their metabolism, their chemical activities transformed into odour. Your scent isn't yours alone. It's a collaborative production, a signature composed by billions of contributors you'll never see. Babies acquire their first microbiomes during birth, inheriting microbial communities from their mothers in what amounts to an invisible christening, a microbial inheritance transmitted alongside genetic inheritance. As a baby passes through the birth canal, it becomes coated with vaginal bacteria, lactobacilli primarily, which colonize the infant's skin, mouth, and digestive system. These are the baby's first microbes, the founding populations of ecosystems that will persist and evolve throughout life. Babies born by caesarean section receive different initial microbes, mostly from skin rather than the birth canal, and studies suggest their microbiomes develop differently, at least initially. Some hospitals now swab C-section babies with vaginal fluids to try to compensate, to provide the microbial inheritance they missed. It speaks to how important we've realized these early colonizers are, how much they shape the developing immune system, the gut environment, even long-term health outcomes. Your invisible inheritance began the moment you entered the world, bacteria from your mother becoming part of you, colonizing the sterile landscape of your newborn body. You were never truly sterile, never existed as a pure human organism without microbial partners, Within hours of birth, you were already a composite, already hosting communities that would help shape your development, already more than just the cells containing your DNA. Antibiotics kill not just disease-causing bacteria, but also beneficial ones, sometimes taking years for communities to recover, leaving scorched microbial earth in their wake. When you take broad-spectrum antibiotics, they don't discriminate. They kill susceptible bacteria indiscriminately, the pathogen making you sick, but also the beneficial species in your gut, on your skin, in your respiratory tract. It's collateral damage, necessary sometimes to save your life, but not without cost. Your gut microbiome can lose diversity during antibiotic treatment, with some species disappearing entirely, others blooming in the disrupted environment. Yet the community structure changes, becomes simplified, sometimes fails to fully recover even months after the antibiotics stop. Some people develop secondary infections like C. difficile colitis because the antibiotics kill the beneficial bacteria that normally keep pathogens in check, creating ecological vacuums that opportunistic species exploit. Modern medicine saves lives with antibiotics, certainly. Infections that would have killed our ancestors are now curable with pills. But we're learning that these drugs are blunter instruments than we thought, that they affect far more than their intended targets. We're trying to develop more selective antibiotics now, drugs that kill pathogens while sparing beneficial species, but it's difficult. Bacteria are biochemically similar enough that finding molecules that distinguish friend from foe remains challenging. So we continue using the tools we have, saving lives while occasionally scorching the microbial ecosystems we depend on, trusting they'll recover, hoping the damage isn't permanent. Some scientists believe our gut microbiome can influence mood, behavior, and even mental health, sending signals to the brain through multiple pathways, chemical, hormonal, neural, that might affect how we feel and think. It's called the gut-brain axis, and we're only beginning to understand how extensive the communication might be between the microorganisms in your intestines and the neurons in your skull. The gut is lined with nerve cells, more neurons than in your spinal cord, sometimes called the second brain. These neurons communicate with the actual brain through the vagus nerve, sending signals about the state of your digestive system. But they might also transmit information about your microbiome, about the chemical environment the bacteria are creating. Some gut bacteria produce neurotransmitters, serotonin, dopamine, GABA, molecules identical to those your brain uses to regulate mood and emotion. Most of your body's serotonin is actually produced in the gut, not the brain. How much of that is made by your own cells versus gut bacteria remains unclear, but the bacteria contribute, they influence the chemical environment, and that might affect how you feel. Studies have shown that transplanting gut bacteria from depressed people into mice can induce depression-like behaviors in the mice. Other studies suggest probiotics might help with anxiety or depression, though the evidence is still preliminary. The boundary between you and your microbiome grows blurrier the more we study it. If gut bacteria influence your mood, your decisions, your mental state, even slightly, then where does your autonomy end and microbial influence begin? It's unsettling, perhaps, but also fascinating. You might be more influenced by your invisible residents than you ever imagined, your thoughts and feelings shaped partly by organisms that aren't you, yet live within you. Fecal microbiota transplants, transferring gut bacteria from healthy to sick people, can cure certain diseases, particularly Clostridium difficile infections that resist antibiotics. Sometimes the cure isn't a chemical we invented, but a community we borrow from someone healthier. Yet a complete ecosystem transplanted from one person to another. The procedure is exactly what it sounds like. Fecal matter from a healthy donor, screened carefully for pathogens, is processed and transferred to a patient's colon, introducing diverse gut bacteria that can outcompete the infection. For recurrent C. difficile infections, fecal transplants are remarkably effective, cure rates above 90%, far better than antibiotics alone. The healthy microbiome establishes itself, restores diversity, fills ecological niches that the pathogen had exploited. Researchers are exploring whether fecal transplants might help with other conditions: inflammatory bowel disease, obesity, autism, depression, conditions we're beginning to suspect might have microbial components. The results are mixed, promising for some conditions, inconclusive for others. But the fundamental insight remains striking. Sometimes healing means not killing the bad, but introducing the good, restoring balance rather than waging war, borrowing ecological resilience from someone whose microbiome functions better than yours. It's humbling in a way. For all our pharmaceutical sophistication, sometimes the best medicine is a healthy person's microbiome, a complex community we still don't fully understand, transplanted as a complete ecosystem, because we can't yet replicate its intricacy through pills or probiotics. The microorganisms in your mouth are so unique they can identify you like a fingerprint, a microbial signature as distinctive as your face, invisible but utterly personal. Everyone's oral microbiome contains similar species. We all have bacteria that colonize teeth, tongues, gums, but the specific strains, their relative abundances, the overall community structure varies from person to person. Forensic scientists can potentially use oral microbiomes to identify individuals, matching the bacterial profile from saliva or bite marks to a suspect. It's more variable than DNA fingerprinting because microbiomes change over time, shift with diet and health and antibiotics. But within reasonable time frames, they're remarkably stable and distinctive. Your mouth hosts a community found nowhere else, shaped by your genetics, your habits, your history, creating patterns that persist for months or years. Even kissing doesn't homogenize oral microbiomes, though partners do share species and temporarily increase microbial overlap. Each person's mouth seems to maintain its characteristic community structure, thus resisting complete takeover by foreign microbes, selecting for species that match their particular oral environment. You carry this invisible signature everywhere, on every cup you drink from, every surface your mouth touches, leaving traces of your unique microbial community like biological breadcrumbs. Certain probiotics are live microorganisms we deliberately consume for health benefits, intentionally adding bacteria or yeast to our bodies in the hope they'll improve our well-being. We've gone from fearing microbes to cultivating them, from sterilizing everything to recognizing that we need microbial partners, that not all bacteria are enemies, that some are allies worth recruiting. The science behind probiotics is still developing. Some strains have solid evidence for specific benefits, certain lactobacilli and bifidobacteria for preventing antibiotic-associated diarrhea, specific strains for managing irritable bowel syndrome. Others are marketed on shakier evidence, promising benefits we haven't conclusively demonstrated. The challenge is that microbiomes are complex, individual, variable. A strain that helps one person might not colonize successfully in another, might not produce the same effects in different ecological contexts. But the fundamental shift in thinking is profound. A century ago, we discovered microbes and learned to kill them with antiseptics and antibiotics. Now we're learning to cultivate them, to select beneficial species, to deliberately introduce them into our bodies. We're recognizing ourselves as ecosystems that need tending, and microbial gardens requiring proper care. It's a more sophisticated understanding, not microbes as universal enemies, but as diverse organisms, some harmful, many beneficial, most neutral, all part of the complex biological landscape we inhabit. Traditional fermented foods contain trillions of microorganisms, making them some of the most microbe-rich things we eat. Living foods teeming with bacteria and yeast that survive cooking and storage to arrive alive in your digestive system. A spoonful of sauerkraut contains more organisms than stars you can see on the clearest night. More living cells than the population of entire countries. Kimchi, kefir, kombucha, miso, traditional pickles. These aren't just flavoured foods. They're living ecosystems, bacterial cultures maintained through fermentation, preserved through acidity and salt, and kept alive until the moment you consume them. Some of these microbes survive your stomach acid and reach your intestines, at least temporarily contributing to your gut microbiome. Whether they colonize permanently or just pass through remains debated, but they interact with your resident microbes, potentially influencing your health through their presence, even briefly. Traditional cultures developed these fermented foods for preservation. Fermentation prevented spoilage in times before refrigeration. But they may have inadvertently been maintaining microbial diversity, regularly consuming live cultures that supplemented their gut ecosystems. Modern commercial versions are often pasteurized, killing the microbes to extend shelf life, eliminating what might have been an important source of microbial exposure. We're rediscovering fermentation now, learning old techniques, cultivating foods our great-grandparents would have considered ordinary but we've nearly forgotten, relearning to eat living foods in a world that's become perhaps too sterile. Some archaea thrive in near-boiling acidic springs with pH levels similar to battery acid, living in conditions that would dissolve your flesh in minutes. They don't just survive in these hellscapes, they prefer them, finding our moderate world inhospitable, too cold, too neutral, too gentle for their tastes. These thermoacidivales have evolved to thrive where chemistry becomes violent, where water bubbles and steams, where dissolved minerals create acidity that would strip paint from metal. Places like Yellowstone's hot springs, volcanic vents in Iceland, geothermal fields across the Pacific Rim. These environments glow with vibrant colours, reds and yellows and oranges and greens, and those colours are living organisms, microbial communities tinting the water with their pigments. The archaea living there have membranes made from different lipids than our cells use, structures that remain stable at temperatures that would cook normal cells. Their proteins fold differently, maintaining function in heat and acid that would denature our proteins instantly. To them, our world is frozen and bland. Room temperature is freezing. Neutral pH is a chemical extreme. They've adapted so thoroughly to their harsh environments that they cannot survive outside them, cannot compete in gentler conditions where mesophilic organisms dominate. They're exiles in their own thermal kingdoms. See, thriving in niches too hostile for anything else remind us that extreme is relative, that what seems uninhabitable to us might be paradise to something else. Microorganisms have been found living 2 miles beneath Earth's surface in solid rock, in the deep biosphere we barely knew existed until recently. Down there in permanent darkness, in rock compressed by unimaginable weight, in temperatures approaching boiling, microorganisms somehow persist. They grow incredibly slowly. Some divide once per century or less, existing in a slow-motion world where time operates differently, where patience is measured in geological epochs. The deep biosphere may contain more biomass than all surface life combined, though it's difficult to measure, difficult even to sample. These organisms live in microscopic pores and fractures in the rock, in thin films of water-coating mineral surfaces, surviving on energy from chemical reactions between rock and water, from radioactive decay of uranium and thorium, from hydrogen gas produced by geological processes. They don't need sunlight, don't need organic matter from the surface. They're completely independent, isolated ecosystems powered by Earth's interior chemistry. Some of these deep subsurface organisms might have been buried when the rock formed, isolated for millions of years, evolving in complete isolation from surface life. They represent hidden biospheres, invisible parallel worlds existing beneath our feet, in the foundations of continents, in ocean floor bedrock, living at Earth's absolute limits for life. We walk above them, never knowing they're there, entire domains of life hidden in stone, patient and persistent, thus outliving everything that dies above them. Tardigrades, microscopic animals often called water bears, can survive the vacuum of space, extreme radiation, temperatures near absolute zero, and pressures 6 times greater than the deepest ocean trenches. They're not microorganisms technically, they're animals with nervous systems, digestive tracts, 8 legs. But they're microscopic, usually less than a millimetre long, and they've mastered survival in ways that seem almost impossible. When conditions become unfavourable, tardigrades enter a state called cryptobiosis. They expel most of their body water, retract their legs, curl into a compact structure called a ton, and essentially die without dying. Their metabolism drops to 0.01% of normal levels, barely detectable. In this state they can survive for decades, possibly centuries, waiting for water to return. When rehydrated, they resume life within hours, unfurling their legs, reanimating as if nothing happened. They've survived exposure to space on the exterior of satellites, endured radiation doses that would kill anything else, tolerated temperatures from minus 272 degrees Celsius to plus 150 degrees Celsius. They produce protective proteins that stabilize their cells during desiccation, antioxidants that prevent damage during cryptobiosis, molecular mechanisms we're only beginning to understand. They're masters of suspended animation, able to pause life almost indefinitely and resume when conditions improve. If humans ever travel between stars, tardigrades might be the only Earth organisms that could survive the journey unprotected, patient seeds waiting centuries to wake in alien worlds. Some bacteria form spores so durable they've survived exposure to space on the exterior of the International Space Station, enduring vacuum, radiation, temperature extremes and UV bombardment for months or years and remaining viable. These bacterial spores are among the most resistant biological structures known, protected by multiple protein coats and specialized molecules that preserve DNA integrity even under assault from cosmic rays. The experiments testing this weren't theoretical. Scientists actually placed bacterial spores on the ISS exterior and retrieved them after prolonged exposure. Many survived. Some were damaged but repairable. A few emerged seemingly unaffected, ready to germinate and resume growth when returned to favourable conditions. It suggests that life could potentially travel between planets on meteorites. that bacterial spores ejected from one world might survive the journey to another, seeding new planets with ancient lineages. This idea, panspermia, life spreading through space, transforms bacteria from earth-bound organisms into potential cosmic pioneers. If rocks blasted from Earth by asteroid impacts can carry viable spores to Mars or Europa or beyond, then life's origin might be older than Earth itself, might trace back to other worlds, other systems. Your bacterial ancestors might be Martian immigrants or travellers from even farther, refugees from a lost world that seeded Earth billions of years ago. It's speculation, but plausible speculation, made possible by the extraordinary durability of bacterial spores tested in the cold emptiness above our atmosphere. Halophils are microorganisms that thrive in salt concentrations that would pickle any other cell, living in environments more saline than the Dead Sea, in evaporating salt lakes where the water is saturated, where salt crystals precipitate from the brine. They've turned osmotic pressure, the force that would normally draw water out of cells until they shrivel and die, into their preferred living condition, evolving strategies to maintain internal balance even in overwhelming salinity. Some halophiles accumulate salt internally, matching external concentrations, becoming essentially bags of brine that maintain structural integrity through specialized proteins that function in high salt. Others produce organic molecules compatible solutes that balance osmotic pressure without interfering with cellular machinery. Both strategies allow them to thrive where nothing else can, in pink salt lakes tinted by their pigments, in salt mines, in brine pools at the bottom of the ocean where water is so salty it doesn't mix with seawater above it, forming distinct boundaries like lakes within the ocean. These environments are sterile to most life. This salt concentration alone is enough to kill bacteria, to preserve organic matter like natural mummification. But halophiles live there comfortably, even requiring the salt, dying in fresh water that would seem hospitable to us. They've claimed ecological niches through extreme specialization, becoming obligate extremophiles that can't survive in moderate conditions, adapted so thoroughly to their harsh homes that they've lost the ability to tolerate anything gentler. Certain fungi can break down toxic compounds in nuclear waste, somehow thriving on radioactive decay, growing on reactor walls in Chernobyl, in contaminated zones where radiation levels remain lethal to humans decades after the accident. They're not just surviving radiation, they seem to be using it, employing melanin pigments to capture energy from gamma rays the way plants use chlorophyll to capture light. The discovery happened almost by accident. Scientists monitoring Chernobyl with robots noticed black fungi growing on the reactor walls, in the most contaminated areas, oriented toward the radiation source as if seeking it out. Testing revealed these fungi grew faster in radiation than without it, that they seemed to harness radioactive energy for metabolism. It's still not fully understood. Radioactive decay shouldn't be usable for biology, shouldn't provide energy in forms cells can capture, yet these fungi apparently do it. They've evolved or adapted mechanisms that turn our most persistent pollution into a resource that potentially offer solutions to nuclear waste storage challenges. Imagine contaminated sites slowly cleaned by fungal action, radiation gradually metabolized and neutralized through biological processes. We're far from deploying such solutions practically, but the fungi exist, proving it's possible, demonstrating once again that evolution finds ways, that life adapts to challenges we create, that the microbial world contains solutions to problems we haven't yet learned to ask properly. Psychrophiles are microorganisms that grow best in freezing temperatures, thriving where ice crystals would shred normal cells, living in glaciers and polar seas and permafrost that remains frozen year-round. They've made homes in cold that would stop most life entirely, evolving antifreeze proteins, flexible membranes, enzymes that function efficiently at temperatures barely above freezing. Some live in sea ice, in the microscopic channels of brine that thread through frozen ocean water, creating tiny liquid habitats within the ice itself. Others inhabit Antarctic ice sheets, growing in thin films of liquid water between ice crystals, surviving on nutrients deposited by wind, on minerals dissolved from dust particles frozen in the ice. They grow slowly. Division might take weeks or months, but they grow, reproducing in conditions that seem inimical to biological processes. These cold-adapted organisms redefine the temperature limits of life, expanding our understanding of where biology can function. They're relevant to astrobiology too. If psychrophiles thrive in Earth's coldest environments, then perhaps similar organisms could inhabit Europa's ice-covered ocean, Enceladus's frozen geysers, the permafrost of Mars. Cold, we've learned, isn't necessarily sterile. It's just another niche, another set of challenges that evolution has solved, another environment where patient organisms have found ways to persist, to metabolize, to maintain the tenuous organized complexity we call life. Some bacteria can survive 1,000 atmospheres of pressure, equivalent to the bottom of the deepest ocean trenches, where the weight of 10 kilometers of water presses down with crushing force that would implode submarines, that would compress air into liquid. These beazophiles, or barophiles, don't just tolerate pressure, they require it, growing poorly or not at all at surface pressures that feel normal to us. The deepest parts of the ocean were once thought lifeless, too dark for photosynthesis, too cold and pressured for biology. But when scientists finally explored the Mariana Trench, they found not sterile water, but ecosystems, fish and amphipods, and bacteria thriving in the crushing darkness miles down. The bacteria living there have adapted their membranes, their proteins, their entire cellular architecture to function under pressure that would denature molecules we depend on. To them, surface pressure is the extreme, the unlivable condition. Bring them up from the deep and they struggle, fail, die in what we consider normal atmospheric pressure. They're as adapted to their high-pressure world as we are to ours, unable to survive outside the deep trenches they call home. It's another reminder that habitability is contextual, that extreme to us is optimal to something else, that life has claimed every niche Earth offers from the highest atmosphere to the deepest abyss. Microorganisms called chemolithotrophs can extract energy from bare rock, essentially eating stone, surviving without any organic matter, without sunlight, on chemistry alone. They oxidize minerals, iron, sulfur, manganese, using the electrons released during these reactions as energy, building their cells from carbon dioxide and minerals dissolved in water. They're the ultimate minimalists, requiring nothing but rock, water, and carbon dioxide to survive. These organisms live in caves, in deep subsurface environments, within mine tailings, anywhere minerals meet water. Some contribute to ore formation, concentrating metals through their metabolism, creating economic deposits through biological action over geological time. Others slowly dissolve rock, contributing to weathering, creating soil from stone through metabolic processes measured in centuries. They represent a completely different energy economy than the one we're familiar with. We depend on sunlight, either directly or through eating organisms that ate plants that photosynthesized. But chemolithotrophs have bypassed the sun entirely, found energy in Earth's crust, in chemical gradients between rocks and water, in oxidation reactions that release tiny amounts of energy they capture with elegant efficiency. They prove life doesn't need stars, doesn't require the organic world, can persist on geology alone. If life exists deep in Mars's crust or in subsurface oceans of icy moons, it might look like this. Patient chemolithotrophs eating rock, independent of sunlight, persisting in darkness on mineral chemistry alone. Certain bacteria produce compounds that work as natural sunscreen, protecting them from UV radiation that would kill most life, molecules that absorb harmful wavelengths and dissipate the energy safely as heat. We're now studying these molecules for human sunscreens, learning from microscopic radiation survivors that have been protecting themselves from solar UV for billions of years. The bacteria produce these compounds, mycosporin-like amino acids, cytonimin, others, not for our benefit but their own, as defenses against DNA damage from sunlight. They live in exposed environments, on rock surfaces, as in shallow water, in desert soil, where UV exposure is intense and constant. The molecules they evolved work remarkably well, absorbing across the UV spectrum, remaining stable under light exposure, protecting cellular machinery from radiation damage. Researchers are extracting these compounds, synthesizing them, testing them as environmentally friendly sunscreen ingredients that might replace chemicals that damage coral reefs and accumulate in ecosystems. It's another case of learning from organisms that solved problems millions of years before we encountered them, of borrowing molecular innovations refined by evolution. The bacteria don't know they're contributing to human welfare. They're just surviving, using chemistry to shield themselves from a hostile sun. And we're clever enough, or humble enough, to recognize their solutions as applicable to our own challenges. Scientists have discovered microorganisms in cloud droplets, living in the atmosphere itself, suggesting that life actively cycles through the sky in ways we're only beginning to understand. Bacteria and fungi drift in air currents, colonize water droplets in clouds, potentially even influence weather patterns and rainfall through their metabolic activities and ice nucleation properties. Some bacteria produce proteins that act as nucleation sites for ice crystal formation, essentially seeding clouds, encouraging precipitation. When these bacteria are abundant in clouds, they might affect when and where rain falls, how clouds form and disperse. And they're not just passive passengers blown aloft by wind, they're active participants in atmospheric processes, living and metabolizing in cloud droplets, reproducing before falling back to Earth in rain. The atmosphere becomes another ecosystem when you consider it this way, not empty space between ground and space, but a habitat, thin and challenging certainly, but colonized nonetheless. Microorganisms lifted from soil or ocean surfaces enter clouds, survive days or weeks in this aerial realm, potentially travel thousands of miles before descending in rain somewhere distant from their origin. They're writing themselves into the water cycle, into climate patterns, into the very air, contributing to processes we thought were purely physical but turn out to have biological components, clouds made more complex by the invisible life they contain. The Human Microbiome Project has identified thousands of bacterial species living in and on us, most previously unknown to science, cataloguing the inhabitants of our bodies like explorers mapping undiscovered countries. We're still discovering our own residents, still learning which species colonize which body sites, what they do there, how they interact with each other and with our own cells. The project revealed just how individualized our microbiomes are. Two people's gut bacteria might differ as much as their faces, similar overall patterns, recognizable types, but unique in details. Some core species appear in nearly everyone. Others are person-specific, shaped by genetics, diet, geography, life history. We each harbour species found nowhere else. These strains unique to our particular bodies, evolved and adapted within our personal ecosystems. It's humbling to realise how much of ourselves we didn't know. These weren't invaders or parasites. They were normal residents, integral parts of human biology. Yet we'd never catalogued them, never understood their diversity or functions. The project continues, expanding beyond bacteria to viruses, fungi, archaea, building ever more detailed maps of the microbial nations that call us home. We're discovering ourselves by discovering them, learning that understanding human biology requires understanding microbial biology, that the boundary between human and microbiome is far more porous and cooperative than we ever imagined. CRISPR gene editing technology was discovered by studying how bacteria defend themselves against viruses, their bacterial immune system repurposed into one of the most powerful genetic tools humans have ever developed. The revolution in genetic engineering came from bacterial immunity, from ancient wisdom we've barely begun applying to our own purposes. Bacteria face constant viral assault. Bacteriophages, viruses that infect bacteria, are everywhere, vastly outnumbering their hosts. So bacteria evolved defenses, including CRISPR systems that remember previous viral infections, storing fragments of viral DNA in their own genomes as a kind of molecular memory. When the same virus attacks again, the bacterium recognizes it and deploys molecular scissors, Cas proteins, that cut the viral DNA into harmless fragments. Scientists realized they could hijack this system, reprogram it to cut any DNA sequence they specified, near turning bacterial immunity into a precision editing tool. Now we use CRISPR to modify crops, to study gene function, to develop treatments for genetic diseases, to potentially edit human embryos. All of it based on how bacteria remember and defend against viruses, on molecular machinery refined over billions of years of bacterial viral warfare. The bacteria weren't trying to help us. They were just surviving, developing immune memories, and we were observant enough to recognize the broader applications of their defensive strategies. Some bacteria can remember previous encounters with viruses and mount faster defenses when attacked again. A primitive immune memory that challenges our assumptions about what requires a brain, about what constitutes learning. Single cells can learn from experience, can adapt their behavior based on past encounters, can improve their defenses through something resembling memory. The CRISPR system is the clearest example. Bacteria literally storing viral sequences in their genomes, maintaining archives of past infections they can reference when threatened. But other forms of bacterial memory exist too. Epigenetic modifications that persist across generations, population-level adaptations that spread through communities, behaviours that change based on environmental history. They're not conscious, not thinking in any sense we'd recognise, yet they exhibit adaptive responses based on past experiences, learning at the cellular level. It blurs the line between behaviour and chemistry, between memory and molecular change. When does a chemical modification become memory? When does a cellular response become learning? Bacteria force us to reconsider these questions, to recognize that information storage and adaptive response, the foundations of memory and learning, don't require nervous systems, might be fundamental properties of life itself, emergent from chemistry, complex enough to maintain patterns, to reference past states, to adjust future behavior based on accumulated information. Scientists recently discovered enormous viruses called giant viruses that blur the line between life and non-life, possessing genes for functions we thought only living cells could perform, challenging our definitions of what viruses are, what life is. These viral Goliaths are visible under light microscopes, larger than some bacteria, and their genomes contain hundreds of genes, including some for metabolism, for DNA repair, and functions that typical viruses lack entirely. Traditional viruses are simple, just genetic material wrapped in protein, dependent entirely on host cells for reproduction, lacking any metabolic machinery of their own. But giant viruses possess genes that suggest greater autonomy, more complexity, capabilities that make them seem almost alive. They blur boundaries we thought were clear, existing in a grey zone between cellular life and molecular parasites. Some scientists argue giant viruses represent a fourth domain of life, separate from bacteria, archaea, and eukarya. Others maintain they're still viruses, just unusually complex ones. The debate continues, but the discovery itself is profound. We found organisms that don't fit our categories, that force us to reconsider fundamental definitions. Perhaps living and non-living aren't binary states but endpoints on a continuum, with giant viruses occupying the murky middle, too complex to dismiss as mere molecular machines, yet too dependent on hosts to call truly alive. Microorganisms can survive in hydrothermal vents on the ocean floor, living off chemicals from Earth's interior rather than energy from the sun, proving life needs no star to thrive. These deep-sea vents spew superheated water rich in hydrogen sulfide, methane, metals, toxic compounds to most life but essential nutrients to chemosynthetic bacteria that form the base of vent ecosystems. The bacteria oxidize these chemicals, extracting energy from reactions between the vent fluids and seawater, using that energy to build organic molecules from carbon dioxide. They're primary producers, like plants, but instead of photosynthesis they perform chemosynthesis, capturing energy from geology rather than sunlight. Entire ecosystems depend on them. Tube worms, clams, crabs, fish, all living in perpetual darkness, all ultimately dependent on bacterial chemosynthesis rather than solar energy. Before discovering hydrothermal vents in 1977, we assumed all life on Earth depended on the sun, either directly or indirectly. Vents proved otherwise, demonstrated that Earth's internal heat could support ecosystems, that photosynthesis wasn't necessary, that life could run on geochemistry alone. It expanded our thinking about where life might exist elsewhere, on Europa, Enceladus, other worlds with subsurface oceans heated by tidal forces or radioactive decay. Worlds that never see sunlight yet might host ecosystems powered by geological chemistry, bacterial gardens growing in alien darkness. Researchers have engineered bacteria to produce insulin, transforming diabetes treatment and making it possible for millions of people to manage a disease that was once fatal. We've turned microbes into pharmaceutical factories, reprogrammed their genetics to brew human proteins in fermentation tanks, harnessing their rapid reproduction and simple genetics for medical production. Before recombinant DNA technology, insulin came from pigs and cows, extracted from pancreases obtained from slaughterhouses, expensive and sometimes causing allergic reactions. But in 1978, scientists inserted the human insulin gene into E. coli bacteria, creating living factories that produced human insulin identical to what your pancreas makes. The bacteria reproduce quickly, express the gene continuously, whereas produce insulin that can be harvested and purified at industrial scale. It was revolutionary, both medically and conceptually. We were no longer just studying bacteria or killing them with antibiotics. We were reprogramming them, editing their genomes to serve human purposes, creating biological manufacturing that's now standard for countless medicines. Bacteria produce growth hormone, clotting factors, vaccines, enzymes, all from engineered genes we've inserted into their genomes. They've become our servants in a sense, domesticated at the genetic level, though they don't know or care about the proteins they're producing, just following genetic instructions we've written into their DNA. Some bacteria can conduct photosynthesis using near-infrared light invisible to our eyes, harvesting wavelengths we can't see, and expanding the spectrum of energy life can use beyond what plants capture. Most photosynthesis uses visible light, red and blue primarily, but certain bacteria have evolved pigments that absorb in the near-infrared, capturing lower energy photons that most photosynthetic organisms ignore. This matters because it expands the range of environments where photosynthesis can occur. Near-infrared penetrates deeper into water, filters through dense microbial mats, where visible light gets absorbed by surface layers. By using longer wavelengths, these bacteria can photosynthesize in shadows, beneath other organisms, in conditions too dim for conventional photosynthesis. They've found unused portions of the solar spectrum and evolved machinery to exploit them. It also matters for astrobiology. If Earth bacteria can photosynthesize using near-infrared, then perhaps life on planets orbiting red dwarf stars, the most common stars in the galaxy, could use similar strategies. Red dwarfs emit mostly infrared rather than visible light. Planets orbiting them would receive dim, reddish illumination we'd find inadequate, but bacteria adapted to infrared photosynthesis might thrive there. Life's energy requirements might be more flexible than we thought, adaptable to different stellar spectra, to dimmer or redder light than Earth receives, expanding the range of potentially habitable worlds. Scientists discovered a bacterium, Candidatus promethearchaeum, that may represent the ancestor of all complex life, a living fossil that might be our great-great-grandmother 1 billion times removed. Evolution's missing link between simple prokaryotic cells and complex eukaryotic ones like ours. It's an archaeon with unusual projections, tentacle-like structures extending from its cell surface, and it lives in deep-sea sediments in symbiosis with bacteria. The discovery is significant because it supports theories about how eukaryotic cells evolved. The leading hypothesis suggests an archaeon engulfed bacteria that became mitochondria, creating the first eukaryotic cell through this merger. Promethearchaeum seems to be doing something similar. Its projections embrace bacteria, forming close associations that might represent early stages of the process that led to us. It's strange to think about our ancestry extending back to single cells in ocean mud, to archaeal organisms that existed before animals, before plants, before fungi, before any complex life. But that's where we come from, ultimately from prokaryotic ancestors that merged and cooperated, that formed partnerships so intimate they became inseparable, creating the cellular complexity that eventually allowed consciousness, thought, the ability to contemplate our own microbial origins. Prometheoarchaeum is a window into that deep past, a glimpse of the world before complexity, when the future of life was still being negotiated through symbiosis in the ancient ocean. Microorganisms on the International Space Station evolved differently than their Earth-based relatives due to microgravity, cosmic radiation, and the unique stresses of the space environment. Space creates new selection pressures, favouring different traits than Earth environments do, potentially birthing the first truly extraterrestrial life forms we've made. It evolved in conditions our planet never provides. Studies of bacteria aboard the ISS show they develop thicker biofilms, altered antibiotic resistance, changes in virulence, mutations that accumulate faster than on Earth. The mechanisms aren't fully understood. Is it the radiation? The microgravity affecting how cells divide? The closed environment of the station? Probably all of these, creating selection pressures bacteria have never encountered in their evolutionary history. This matters for long-duration spaceflight, for potential colonization of other worlds. The bacteria travelling with astronauts will evolve, adapt to space conditions, become different from their Earth ancestors. After generations aboard a Mars colony, or a generation ship travelling to another star, human microbiomes might diverge from Earth's, creating new strains shaped by alien environments. We're watching the beginning of this process on the ISS, seeing evolution accelerated by novel conditions, witnessing the birth of space-adapted life, microorganisms becoming extraterrestrial through human-created environments orbiting above the planet that spawned them. If we find life on Mars, it will almost certainly be microbial, single cells clinging to existence underground, in ice, in pockets of liquid water, kilometers beneath the frozen surface where sunlight never reaches, and temperatures hover just above freezing. First contact won't be with aliens bearing messages or advanced civilizations. It will be with Martian bacteria, organisms that might not even notice we exist. Too simple for awareness, just chemistry organized in patterns that persist and replicate. Mars was once warmer, wetter, more hospitable. Rivers flowed across its surface billions of years ago. If life arose during that ancient epoch, it might have retreated underground as the planet dried and froze, surviving in subsurface refuges where liquid water persists, where geothermal heat maintains habitable conditions. These Martian microbes, if they exist, would have been isolated for billions of years, evolving independently from Earth life, potentially representing a second genesis, a separate origin of biology. Finding them would answer one of humanity's deepest questions. Are we alone? Even simple bacteria on Mars would suggest life is common. The chemistry tends toward biology given the right conditions, that the universe might be teeming with microorganisms even if complex life remains rare. Yeah, first contact might happen not through radio telescopes listening for distant signals, but through rovers drilling into Martian ice, microscopes examining soil samples, discovering cells whose ancestors never knew Earth, alien microorganisms that evolved under a different sun. Earth's microorganisms could potentially survive the journey to Mars on ejected meteorites, riding rocks blasted into space by asteroid impacts, crossing the void between planets in stone vessels, seeding neighbours across the cosmic distance. When large asteroids strike Earth, They excavate material with such force that rocks escape the planet's gravity entirely, some eventually crossing paths with Mars and falling there as meteorites. Scientists have found Martian meteorites on Earth, rocks proven to be from Mars by their isotopic signatures, were ejected by impacts millions of years ago and eventually captured by Earth's gravity. If rocks can travel from Mars to Earth, the reverse is possible, and if bacterial spores can survive embedded in rock, shielded from radiation and vacuum, then Earth life might have already contaminated Mars, might have been seeding the red planet for billions of years. This is panspermia at planetary scale, life spreading not across galaxies but between neighbouring worlds, hitching rides on impact ejector. It means Mars and Earth might share ancestry. That Martian life, if it exists, might be our distant cousins, descended from common ancestors launched into space when both planets were young and impacts more frequent. Or perhaps we're the Martians, Earth life descended from microorganisms that originated on Mars and arrived here on meteorites. Refugees from a dying world that found refuge on a more hospitable neighbour. We don't know. But the rocks have travelled. The possibility exists. Life might be more mobile between worlds than we ever imagined. Europa, Jupiter's moon, likely has more water than Earth beneath its ice shell. A global ocean, perhaps 100 kilometres deep, warmed by tidal forces from Jupiter's immense gravity. It's the solar system's largest ocean, and it has never seen sunlight. Locked beneath ice perhaps 10 to 30 kilometres thick, a dark sea that might nonetheless host microbial life. The ocean is kept liquid by tidal heating. Europa's elliptical orbit causes Jupiter's gravity to flex and squeeze the moon rhythmically, generating friction, creating heat. That heat might support hydrothermal vents on Europa's ocean floor, similar to Earth's deep-sea vents, providing chemical energy for chemosynthetic bacteria. Everything needed for life might be there: liquid water, chemical energy, organic compounds delivered by comets, billions of years for chemistry to organize into biology. We've sent probes to Jupiter, flown past Europa, analyzed its surface ice. Future missions plan to land, to drill through the ice if possible, to send submarines into that alien ocean. What we find there, if we find anything, will be microorganisms, chemosynthetic bacteria feeding on vent chemicals, archaea adapted to cold and pressure and perpetual darkness, ecosystems that never knew a star, life proving again that biology needs no sun, just chemistry and liquid water and time. The shadow biosphere hypothesis suggests Earth might host microbial life so fundamentally different from known life that we haven't recognized it yet. Organisms using different biochemistry, different genetic systems, different solutions to the problem of living. Aliens might already live among us, invisible because we've only looked for life like the life we know, because our detection methods assume all life uses DNA and RNA and the same amino acids and sugars. What if some microorganisms use different nucleic acids, different proteins, different membrane chemistry? They might be sitting in soil samples we've analysed, dismissed as chemical noise, because they don't show up in DNA tests, don't grow in our culture media, don't match patterns we search for. We might be surrounded by weird life. A parallel biosphere that originated separately or evolved so divergently from conventional life that we don't recognize it as living. It's speculative, certainly. We have no confirmed examples. But absence of evidence isn't evidence of absence, especially when our search methods have blind spots. Life might be more diverse than we realize, exploring more solutions to biochemistry than we've encountered, existing at the edges of detectability because it's too strange to recognize. Perhaps in desert varnish, in acid mine drainage, in deep subsurface rock, weird microorganisms persist, unnoticed because they don't fit our templates for what life should look like. Amino acids, life's building blocks, the molecular components of proteins, have been found in meteorites, in space-returned samples, in the clouds of interstellar space, suggesting that chemistry favours biology's emergence, that the universe seems strangely eager to assemble the pieces that become life. These aren't simple molecules, they're complex organic compounds that form readily in space environments, in asteroids, in stellar nebulae, wherever carbon and nitrogen and hydrogen mix in the presence of energy. The Murchison meteorite, which fell in Australia in 1969, contained over 70 different amino acids, only 8 of which are used by Earth life. The rest were variations, alternative configurations that biology here never adopted, but that chemistry readily produces. It suggests amino acid formation is common, perhaps inevitable given the right conditions, that the molecular alphabet of proteins emerges naturally from chemistry without requiring biology to create it. This has profound implications. Now, if the building blocks of life form spontaneously throughout the universe, if they rain down on planets in meteorites, if they're present in comets and asteroids, then perhaps life's emergence is likely rather than rare, probable rather than miraculous. The raw materials are everywhere, scattered through space waiting for liquid water and energy and time to organize into something living. We might exist not because chemistry accidentally stumbled into life, but because chemistry inevitably moves toward complexity, toward organization, toward patterns we recognize as biology. Microorganisms might survive interstellar travel inside comets, potentially spreading life between star systems, riding icy bodies across light years of emptiness, hibernating in frozen time until captured by new solar systems billions of years later. Thus, comets are time capsules, ice and rock and organic molecules preserved since the solar system's formation, travelling through space for eons, occasionally captured by planetary gravity. If bacteria can survive frozen in Antarctic ice for millions of years, if they can endure radiation and vacuum as spores, then perhaps they could survive inside comets, shielded by ice from cosmic rays, dormant but viable across interstellar distances. Comets ejected from one solar system might eventually encounter another, falling onto planets as they did during Earth's early bombardment, delivering not just water and organic molecules, but living organisms ready to colonize new worlds. This is panspermia at galactic scale, life as a cosmic phenomenon, spreading through space on natural vehicles, seeding multiple star systems from common sources. It would mean Earth life might have relatives light years away, microbial cousins descended from the same comet-born ancestors, evolution proceeding in parallel on separate worlds. We'd be part of a biological network spanning the galaxy, connected through deep time by wandering comets carrying frozen passengers between stars, life spreading slowly but inexorably through the cosmos on ice and time. Saturn's moon, Enceladus, shoots gazers of water into space through cracks in its ice shell, plumes that contain organic molecules, salts, and evidence of hydrothermal activity. We're flying through the spray of what might be an inhabited ocean, spacecraft sampling potential alien life expelled directly from a subsurface sea, collecting samples without even landing. The Cassini spacecraft flew through these plumes multiple times while analysing their composition. The water contains silica particles that form only at high temperatures, suggesting hydrothermal vents. It contains complex organic molecules, hydrogen gas, chemical energy sources. Everything needed for life seems present: liquid water, chemical energy, organic compounds, stability over billions of years. Somewhere beneath Enceladus's frozen surface, a warm ocean might host ecosystems, chemosynthetic bacteria feeding on vent chemistry, archaea adapted to the moon's cold darkness. Future missions might sample these plumes more thoroughly, searching for amino acids, for cellular structures, for any hint of biology in the expelled ocean water. We wouldn't need to drill through kilometers of ice or land on the surface. The ocean is telling us its contents directly, fountain-like, with shooting samples into space for us to collect. If life exists there, we might detect it from orbit, might discover inhabited oceans on other worlds without ever touching them, finding microorganisms in geysers mist, in frozen droplets that tell the story of dark seas we've never seen. The search for extraterrestrial intelligence focuses on radio signals, on messages from technological civilizations, but biosignatures from microorganisms might be easier to detect, more persistent, more likely to reveal inhabited worlds. Alien civilizations might rise and fall, might exist briefly or never develop technology, but their microbes could persist for billions of years, altering atmospheres in detectable ways, producing chemical signatures visible across interstellar distances. Oxygen in Earth's atmosphere is a biosignature. It shouldn't persist at current levels without continuous biological production. Methane combined with oxygen is even more suggestive, a chemical disequilibrium that points toward life. Future telescopes analysing exoplanet atmospheres will search for these signatures, for atmospheric compositions that require biological explanation, for planets where chemistry seems driven by life rather than geology alone. We might find inhabited worlds not by receiving their broadcasts, but by detecting their microbes' waste products, oxygen and methane, and other gases produced by photosynthesis and metabolism, chemical fingerprints of invisible ecosystems breathing on distant planets. The first confirmed extraterrestrial life might be microbial, detected spectroscopically light years away. Near organisms we'll never sample directly, but whose presence alters their world's atmosphere enough to announce, life exists here. The universe might be full of microbial worlds, planets where life never evolved beyond single cells but still thrives, still transforms atmospheres, still writes itself into planetary chemistry in ways we can detect from across the galaxy. If we terraform Mars, microorganisms would likely be the first colonists, the pioneers we'd send ahead, preparing the ground for everything else. They'd begin the work of transformation, processing regolith into soil, producing oxygen, fixing nitrogen, breaking down perchlorate salts, establishing ecosystems that more complex organisms could later join. We might release cyanobacteria in subsurface ice caves, photosynthetic organisms producing oxygen in protected environments. We might introduce nitrogen-fixing bacteria to develop soil fertility, decomposers to process waste, extremophils adapted to Mars's harsh conditions, radiation, cold, low pressure, toxic soil chemistry. These microbial pioneers would work for decades or centuries, slowly changing local environments, creating pockets of habitability that could gradually expand. Humanity's expansion to the stars begins not with astronauts planting flags, but with invisible pioneers we'll send ahead. Bacterial colonies establishing themselves in alien soil. Archaea adapted to process Martian chemistry. Microorganisms beginning the patient work of making other worlds livable. They won't know they're serving human purposes. They'll just be surviving, reproducing, doing what life does, spreading, adapting, transforming environments. But through their metabolism, through their persistence, they'll prepare worlds for our arrival. Microbial ambassadors carrying Earth life to other planets. Some scientists believe early Earth microorganisms arrived from space, that life didn't originate here but elsewhere, making us all descendants of alien bacteria. Our family tree extending beyond this planet to interstellar clouds that seeded multiple worlds. It's called panspermia, and while it doesn't solve the origin of life question, it just moves it elsewhere. It transforms our understanding of our place in the cosmos. If Earth life descended from organisms that arrived on comets or meteorites, then we're not native. We're colonists, immigrants from some other world or interstellar environment, refugees who found haven here billions of years ago. Our microbial ancestors might have originated in stellar nebulae, he says, in the atmosphere of a failed star, on an ancient planet that no longer exists, travelling through space until captured by young Earth's gravity. This would mean life might be older than Earth itself, that biology might predate our planet's formation, that the universe might have been experimenting with life long before our world existed. It would connect us to larger cosmic processes, make us participants in life's spread across the galaxy, rather than an isolated occurrence on one planet. Your lineage might trace back not just to Earth's first cells, but to something older, something interstellar, ancient beyond our planet's history. We might all be aliens in the deepest sense, carrying genetic memories of worlds and spaces we've never known, descended from travellers who crossed the dark between stars before finding Earth. Bacteriophages, viruses that kill bacteria, might become crucial weapons against antibiotic-resistant infections, offering solutions to one of medicine's most pressing crises. The enemy of our enemy might save us, viral assassins that target only specific bacteria, leaving human cells and beneficial microbes untouched. They're exquisitely selective. Each phage evolved to attack particular bacterial species, sometimes particular strains, using molecular recognition systems honed over billions of years of bacterial viral warfare. Phage therapy was developed in the early 1900s, before antibiotics, abandoned in the West when penicillin proved easier to manufacture and store. But in former Soviet countries, phage therapy continued, refined, creating libraries of therapeutic viruses targeting different pathogens. Now, as antibiotic resistance spreads, as bacteria evolve defenses against our most powerful drugs, we're reconsidering phages, rediscovering abandoned weapons in our war against infection. Phages evolve alongside bacteria, constantly adapting to overcome bacterial defenses, engaged in evolutionary arms races that continue whether we participate or not. When bacteria develop resistance to antibiotics, we must develop new drugs, a slow process limited by human ingenuity. But when bacteria develop resistance to phages, the phages evolve countermeasures naturally, automatically, maintaining effectiveness through evolution's relentless optimization. We might not need to invent new weapons, we might just need to harness nature's ongoing warfare, recruiting viral warriors already adapted to killing the bacteria threatening us. Your immune system is largely trained by exposure to harmless microorganisms in childhood, educated through contact with diverse bacteria that teach your defenses which threats are real and which are benign. Growing up too clean might leave your immune system uneducated, untrained, prone to mistakes like attacking harmless pollen or food proteins instead of actual pathogens. It's called the hygiene hypothesis, the idea that reduced microbial exposure in developed countries contributes to rising allergies, asthma, autoimmune diseases. Children raised with pets, on farms, in larger families tend to have fewer allergies, possibly because they encounter more diverse microorganisms that properly calibrate their developing immune systems. The immune system needs practice, needs to encounter microbial diversity to learn discrimination, to develop appropriate rather than overactive responses. This doesn't mean hygiene is bad, that we should abandon sanitation. We've eliminated many diseases through cleanliness, saved countless lives through sewage treatment and water purification, but perhaps there's a balance, a middle ground between pathogen exposure that kills and sterile environments that leave immune systems untrained. Perhaps we need some microbial diversity, some exposure to harmless organisms that educate rather than sicken, that train our defenses without causing disease. The challenge is finding that balance, providing immune education without risking infection, exposing children to beneficial microbes while protecting them from harmful ones. Some bacteria produce compounds that become antibiotics, chemical warfare molecules we've borrowed for medicine, weapons one microorganism evolved to use against competitors that we've repurposed to save human lives. Penicillin was a fungus's weapon against bacteria, a molecule penicillium produced to kill competitors in its environment, and we discovered it accidentally. Noticed bacterial death around fungal contamination, recognized its medical potential. Most antibiotics come from microorganisms, particularly soil bacteria and fungi competing for resources in densely populated microbial communities. They evolved chemical warfare over millions of years, developing molecules that disrupt bacterial cell walls, interfere with protein synthesis, prevent DNA replication. We've screened thousands of microbial species, searching for useful compounds, isolating natural antibiotics and modifying them chemically to enhance effectiveness. But we're borrowing weapons from an ongoing war. The bacteria being attacked by these compounds have been evolving resistance for millions of years, long before we started using antibiotics medically. We entered an ancient conflict, took one side's weapons without fully understanding the evolutionary dynamics, the endless arms race between microbial attackers and defenders. Now we're learning that this war has no permanent victories, that resistance is inevitable, that we're participants in evolutionary processes far older and more relentless than human medicine. Antibiotic resistance evolves in bacteria so quickly that some drugs become useless within years of introduction. Organisms reproducing thousands of times faster than us, adapting through generations we can barely track. We're in an evolutionary arms race against opponents with enormous advantages, vast population sizes, rapid generation times, horizontal gene transfer allowing them to share successful mutations across species boundaries. A single bacterial infection might contain billions of cells. Most die from antibiotic exposure, but if even one carries a resistance mutation, it survives, reproduces, and within hours its descendants dominate the population. Then that resistance gene can spread to other bacterial species through plasmids, mobile genetic elements transferred between cells, creating resistance that jumps across species faster than we can develop new drugs. The mathematics favour them. We invest years and billions of dollars developing new antibiotics. They evolve resistance through natural selection in months, sometimes weeks. We're locked in a race where our opponents move faster, reproduce quicker, innovate through evolution's trial and error while we depend on slower human research and development. Perhaps the solution isn't winning this race, but changing its terms, using phages that co-evolve with bacteria, developing narrow-spectrum drugs that preserve beneficial microbes, reducing antibiotic use to slow selection pressure, recognizing that complete victory might be impossible in evolutionary conflicts. Certain viruses naturally hunt and destroy cancer cells, leading to experimental treatments using modified viruses as precision medicines, viral scalpels guided by evolution's recognition systems. Some viruses preferentially infect rapidly dividing cells, exactly the characteristic that defines cancer. We're engineering these viruses, we're enhancing their cancer-targeting abilities, using them to deliver genes that trigger immune responses or directly kill tumour cells. Oncolytic viruses, cancer-killing viruses, represent a complete inversion of our relationship with viruses. For centuries, viruses were purely enemies, causes of disease we fought to eliminate. Now we're turning them into allies, weapons against cancer, delivery vehicles for gene therapy. We're reprogramming their targeting mechanisms, their replication strategies, their cellular effects, creating designer viruses that spare healthy tissue while attacking tumours. It's still experimental, still being refined, but some treatments have shown promise. Viruses injected into melanomas replicate in cancer cells, burst them open, release tumour antigens that train the immune system to recognise and attack the cancer. The viruses are doing what they evolved to do, infecting cells, replicating, spreading. But we've aimed them, pointed them at our enemies, turned humanity's ancient adversaries into precision therapeutics, guided by viral biology we're learning to manipulate. The bacteria Helicobacter pylori causes ulcers and stomach cancer, yet also protects against some allergies and asthma, complicating our urge to eliminate it entirely. It colonizes human stomachs, survives in stomach acid that kills most organisms, living in the mucus layer coating the stomach lining. In some people it causes disease, ulcers, gastritis, eventually cancer. In others it lives harmlessly for decades, and some studies suggest its presence protects against certain allergic and autoimmune conditions. It's been with humans for at least 100,000 years, possibly longer. Ancient symbiont that may have once been beneficial or neutral, but becomes pathogenic in modern environments. As sanitation improved and H. pylori prevalence declined in developed countries, allergies and asthma increased. Correlation isn't causation, but the relationship suggests H. pylori might have immunomodulatory effects, that its presence might help calibrate immune responses, preventing overreactions to harmless triggers. Even pathogens can be beneficial in balance, in context, in certain individuals. The bacteria that causes disease in one person might protect another, might serve useful functions we don't fully understand. Complete eradication might have unintended consequences, might remove organisms that, despite their risks, provide subtle benefits we only notice after they're gone. Biology is complex. Most ecosystems are interconnected, and even harmful organisms often play roles we haven't fully appreciated. Scientists are developing living therapeutics, engineered bacteria that detect and treat diseases from inside your body, sensing chemical signals and responding autonomously like microscopic physicians working in your gut. They're designing bacteria with genetic circuits that activate in response to inflammation markers that produce anti-inflammatory compounds when needed, that sense and respond to your body's state without external control. Imagine bacteria engineered to detect early signs of inflammatory bowel disease and release therapeutic proteins locally, directly at inflammation sites. Or bacteria that sense low iron levels and produce molecules enhancing iron absorption. Or bacteria programmed to detect cancer metabolites and activate immune responses. These aren't static medicines. They're responsive, adaptive, biological robots that diagnose and treat continuously, adjusting their behavior based on your body's changing needs. Future medicine might involve cultivating pharmaceutical microbes in your gut like a medicinal garden, maintaining populations of engineered bacteria that provide ongoing health surveillance and treatment. They'd work autonomously, sensing conditions we can't measure without laboratory tests, responding faster than we could manually, providing personalized medicine that adapts in real time to your individual physiology. We're still far from this vision, but the basic technologies exist: genetic circuits, biosensors, engineered organisms that respond predictably to environmental signals. We're learning to program bacteria like computers, writing code in DNA that executes in living cells. Some microorganisms produce neurotransmitters like serotonin and dopamine in your gut, potentially affecting your brain, your mood, your mental state. Your intestines contain neurons, neurotransmitter receptors, signaling molecules identical to those in your brain, creating what scientists call the gut-brain axis. Bidirectional communication between intestinal and neurological systems. Gut bacteria produce enormous amounts of serotonin. Perhaps 90% of your body's total serotonin is synthesized in the gut, much of it by bacteria or influenced by bacterial metabolites. They also produce GABA, dopamine, other neurotransmitters that regulate mood, anxiety, cognition. These molecules might not cross the blood-brain barrier directly, but they influence the vagus nerve, affect immune signaling, alter hormone production, potentially impacting brain function through indirect pathways. Your mood might be partially manufactured by residents in your intestines, invisible chemists brewing emotion through metabolism you don't control and barely perceive. When you feel anxious or depressed, when your mood shifts without obvious cause, bacteria might be contributing, altering neurochemical environments through their metabolic activities. It's unsettling, but also hopeful. If gut bacteria influence mental health, then modulating the microbiome might treat psychiatric conditions, might offer new approaches to depression and anxiety beyond traditional pharmaceuticals. You're just targeting the microbial communities that help regulate your emotional state. Bacteriophage therapy was abandoned in the West when antibiotics emerged, but remained in use in Soviet countries, creating parallel medical traditions that diverged for decades, developing different approaches to treating bacterial infections. While Western medicine relied on antibiotics, Soviet and Eastern European countries maintained phage research institutes, built libraries of therapeutic viruses, treated patients with phages when antibiotics failed. The divergence was partly political. Cold War isolation meant less scientific exchange, but also practical. Antibiotics were easier to standardize, store, distribute. Phages required careful matching to bacterial strains, needed fresh preparations, were difficult to patent. Western pharmaceutical companies invested in antibiotics. Soviet medicine, with different economic incentives, continued developing phages. Now the forgotten path might save us. As antibiotic resistance spreads, Western researchers are rediscovering phage therapy, consulting decades of Soviet research, learning from treatments developed in Georgia, Poland, Russia, while Western medicine pursued different strategies. The parallel tradition preserved knowledge, maintained techniques, continued experiments that Western medicine abandoned. Sometimes the road not taken circles back, becomes relevant again when circumstances change, when old problems return in new forms requiring solutions we once discarded. Your appendix, once thought useless, a vestigial organ serving no purpose, a candidate for evolutionary obsolescence, might serve as a reservoir preserving beneficial gut bacteria after illness, a microbial safe house, a backup copy of your bacterial community stored in an anatomical bunker. When severe diarrhea or antibiotics deplete your gut microbiome, the appendix might help recolonize your intestines, maintaining bacterial diversity through periods of disruption. The appendix is a narrow, dead-end pouch attached to your intestine, easily sealed off from the main digestive tract. Bacteria inside might be protected from being flushed out during diarrhea, shielded somewhat from antibiotics, preserved in this protected compartment. After the crisis passes, they might emerge, reseeding your gut with beneficial species that might otherwise be lost. It's still debated, still being researched, but it reframes appendicitis in interesting ways. Perhaps the appendix serves a real function, particularly important in environments without modern medicine, where bacterial preservation during illness matters more. Modern sanitation, antibiotics, and medical care might have reduced its importance, making appendicitis more dangerous than the organ is useful. But originally, evolutionarily, it might have been genuinely helpful. A reservoir maintaining microbial backup copies, insurance against the catastrophic loss of your gut ecosystem during severe illness. We still don't fully understand why bacteria coordinate their behavior through quorum sensing instead of acting independently. What drives single cells to sacrifice individual advantage for collective benefit in ways that seem mysteriously altruistic? From a purely selfish gene perspective, coordination is puzzling. Why would a bacterium delay beneficial behaviors until neighbors are present? Why tie your success to population density rather than acting immediately for personal gain? Perhaps the answer lies in efficiency. Some behaviors only work at scale. Bioluminescence too dim from one cell to be useful. Biofilm formation requiring collective architecture. Toxin production, wasteful unless enough cells contribute simultaneously. Quorum sensing might be less about altruism and more about conditional cooperation. Cells waiting for conditions where collective action becomes individually beneficial, where the group behavior serves each participant better than solo action would. But it still feels like something more, like coordination that approaches decision-making, in us like cells communicating and adjusting strategies based on social context. They're not thinking, not conscious, yet the outcomes resemble deliberation, cooperation, responses to community that seem sophisticated for organisms without brains, without even nuclei. Perhaps cooperation is more fundamental than we realize, built into biology at the molecular level, emerging not from complex cognition, but from simple rules repeated billions of times, collective behavior arising from chemistry rather than thought. Scientists estimate 99% of microbial species cannot be cultured in laboratories with current techniques, meaning the invisible world is largely unexplored, a dark matter of biology we know exists but cannot touch, cannot study directly. We can detect their DNA in environmental samples, see their genetic signatures in soil and water and air, but we cannot grow them in petri dishes, cannot maintain them in culture, cannot study their metabolism and behaviour with traditional methods. They're called unculturable, though that's probably incorrect. They can be cultured, just not under conditions we've tried yet. Perhaps they require specific chemical signals from other species, need symbiotic partners we haven't identified, depend on conditions too specific or complex for us to replicate. Each culturable species we know might represent dozens of unculturable relatives we've never isolated, never grown, never examined beyond their genetic shadows in environmental DNA. It's humbling. We've studied microorganisms for over 3 centuries, yet the vast majority remain unknown, uncharacterized, mysterious. We can sequence their genomes from mixed samples, infer their metabolic capabilities from genes, predict their ecological roles from DNA alone, but we cannot see them divide, cannot watch them interact, cannot experiment with them directly. The microbial world extends far beyond what we've accessed, vast territories of diversity we've barely begun exploring, organisms that might revolutionize biotechnology or medicine if only we could coax them into growing in our laboratories. We don't know if viruses should be classified as living organisms or complex molecular machines, whether they deserve placement on the tree of life or exist in some other category entirely. The boundary between chemistry and biology blurs in viruses, forcing us to reconsider what alive actually means, what criteria separate living from non-living. Viruses lack metabolism. They don't eat, don't breathe, don't generate their own energy. They cannot reproduce independently. They require host cells to replicate. They're essentially genetic information wrapped in protein, inert until they encounter compatible cells. By most definitions of life, homeostasis, metabolism, growth, reproduction, response to stimuli, viruses fail multiple criteria. Yet they evolve, they adapt, they replicate, they interact with living systems in sophisticated ways. Giant viruses complicate the picture further, possessing genes for functions we thought required cellular life. Where exactly does the line fall? Is life defined by having metabolism, by independent reproduction, by cellular structure? Or is it something more fundamental, information that perpetuates itself, patterns that persist through time, your organization that resists entropy? Viruses force us to examine our definitions, to recognize that perhaps living isn't a binary classification but a spectrum, and viruses occupy the ambiguous middle ground, too complex to dismiss as mere chemistry, yet too simple to call fully alive. The origin of the first self-replicating molecule remains unknown. Chemistry's transformation into biology is still mysterious. The transition from lifeless matter to living systems still poorly understood despite decades of research. We've traced evolution back nearly to the beginning, following genetic lineages to the last universal common ancestor. But the first step from non-life to life continues to elude us, hidden in deep time before any fossils formed. How did non-replicating molecules become replicating ones? How did metabolism arise? How did cellular membranes form? We have hypotheses, RNA world scenarios where RNA acted as both information storage and catalyst, metabolism first theories where chemical cycles preceded genetics, various proposed pathways from chemistry to biology, but no consensus, no definitive answer, just competing ideas about how the transition might have occurred. Perhaps it happened many times, failed countless ways before succeeding. Perhaps it required specific conditions we haven't identified or cannot replicate. Perhaps it was unlikely, a rare accident that happened to occur on early Earth. Or perhaps it's inevitable given the right chemistry and energy. We don't know. The origin of life remains one of science's deepest mysteries. The moment when chemistry learned to perpetuate itself, when matter became alive, when the universe became capable of contemplating its own existence through biological organisms that emerge from purely physical processes we're still trying to understand. Some microorganisms can repair catastrophic DNA damage perfectly, rebuilding their genetic code from hundreds of fragments with precision that seems almost impossibly accurate, editing errors from the blueprint of life with molecular proofreading abilities that make our cellular repair mechanisms look primitive by comparison. Deinococcus radiodurans can survive radiation that shatters its genome into thousands of pieces, and within hours it reassembles perfect copies, virtually error-free, through mechanisms we don't completely understand. How do they do it? They maintain multiple genome copies, providing templates for repair. They have extraordinarily efficient wrecker proteins that align DNA fragments, searching for homology, matching pieces like molecular jigsaw puzzles. They possess repair enzymes that work with astonishing fidelity, stitching fragments together accurately even when the genome is so damaged it seems irreparable. The entire cell coordinates repair, prioritizing genome reconstruction over everything else, working tirelessly until integrity is restored. It challenges our understanding of biological limits, of information preservation, of how accurately cells can maintain genetic information despite damage. These bacteria possess molecular editing capabilities we're trying to understand and perhaps harness. Repair systems refined over evolution in high radiation environments, solutions to DNA damage that might inform cancer treatment, radiation protection, preservation of genetic information in extreme conditions. They're molecular librarians of extraordinary competence, maintaining perfect archives despite catastrophic damage, proving that life's information can be remarkably resilient when protected by the right molecular machinery. We're uncertain whether Earth's microbiomes are changing faster due to human activity than they can adapt, whether we're conducting an unprecedented experiment on invisible ecosystems we barely understand with potentially irreversible consequences. Antibiotics, pollution, climate change, habitat destruction all affect microbial communities in ways we're only beginning to measure, creating selection pressures that might be pushing ecosystems beyond their adaptive capacity. Ocean acidification alters marine microbiomes. Agricultural chemicals change soil bacteria. Antibiotics in wastewater select for resistance genes. Warming temperatures shift species distributions. Each change cascades through microbial networks, affecting nutrient cycles, affecting larger organisms that depend on microbial partners, potentially destabilizing ecosystems in ways we won't recognize until damage becomes obvious. The challenge is that we lack baseline data. We don't know what normal microbial diversity looked like before industrial civilization, before antibiotics, before modern agriculture. We're documenting changes but can't always compare them to original states. And microbiomes are complex, interconnected, their responses to perturbation often non-linear and unpredictable. We might be pushing them toward tipping points, thresholds beyond which they reorganize into different stable states, degraded states, less diverse states, states less able to support the ecosystems we depend on. Or perhaps they're adapting, evolving, more resilient than we fear. We genuinely don't know. We're changing the invisible world faster than we can study it. The question of whether microorganisms have any form of awareness or sensing beyond chemical reactions remains philosophical, blurring the boundaries between mechanism and experience even in the smallest living things. Where does mechanism end and experience begin? Do bacteria sense their environment or merely respond mechanistically? Is there any subjective experience, however primitive, in a single cell, or is it purely chemistry with no inner life whatsoever? They respond to stimuli, moving toward nutrients, away from toxins, changing gene expression based on environmental conditions. They make decisions in computational terms, following gradients, choosing between alternative metabolic pathways, coordinating with neighbors. But is any of this accompanied by experience, by any form of sensation, or is it entirely unconscious, mechanical, just molecules interacting according to physical laws with no awareness anywhere in the system? We don't know. We probably can't know, at least not with current methods. Consciousness and subjective experience are difficult enough to confirm in animals, nearly impossible to assess in organisms without nervous systems. Perhaps awareness requires certain complexity we can define. Perhaps it's a continuum, with bacteria possessing some infinitesimally small degree of sensation. Or perhaps there's a hard threshold and single cells fall entirely below it. The question sits at the intersection of biology and philosophy, unanswerable but worth contemplating, asking where experience enters the universe, whether life always comes with some form of interiority, or whether awareness is a later evolutionary innovation absent from biology's simplest forms. Scientists debate whether the vast diversity of microbial life we see is ancient or relatively recent, whether the explosion of bacterial and archaeal lineages occurred billions of years ago or more recently in geological time. The timeline of invisible evolution might span the planet's entire history, or might be surprisingly compressed, with much of current diversity arising in the last billion years, rather than throughout life's 3.5 billion year tenure on Earth. Molecular clocks, estimates based on mutation rates, give different answers depending on assumptions about calibration and evolutionary rates. Some analyses suggest microbial lineages diverged early, ancient splits creating the diversity we see today. Others suggest more recent radiations, bursts of diversification in response to environmental changes like oxygen accumulation or snowball Earth events, or the rise of eukaryotes creating new ecological opportunities. The uncertainty reflects how difficult it is to date events without fossils, to infer timelines from genetic differences when we don't fully know how quickly mutations accumulate, how selection and drift affect molecular evolution in different lineages. The deep history of microbial life remains partly opaque. Events billions of years past, leaving ambiguous signals in contemporary genomes. We know microorganisms are ancient, but the pattern of their diversification, steady and gradual or punctuated by rapid radiations, remains debated. The shape of evolution's tree in its invisible branches still being determined through genetic archaeology and mathematical modelling. We don't know if life necessarily begins microscopically, or if Earth's pattern represents just one possible path among many alternatives. Perhaps somewhere else life began large and evolved smaller, inverting our assumptions about biology's arc, complexity preceding simplicity in configurations we've never imagined. On Earth, simple preceded complex, microscopic preceded macroscopic, prokaryotes preceded eukaryotes. But is this inevitable? Could chemistry organize into large structures first, simplifying later? Could life start as macroscopic crystals or complex polymers that later fragmented into cellular forms? Could there be evolutionary pathways we've never conceived because we've only observed one example, one data point of how chemistry becomes biology? Perhaps Earth's pattern is universal, dictated by physics and chemistry in ways that always favor small, simple beginnings. Or perhaps it's contingent, one possibility among many, and alien life might follow completely different trajectories toward complexity. We won't know until we find other examples, other biospheres that developed independently, that might show us alternative paths from chemistry to biology, from non-life to life. Earth has shown us one way. Whether it's the only way, or merely the way it happened here, remains an open question we can't answer from a single example, from one world's unique history of biological emergence and elaboration. The ultimate question remains, are microorganisms common throughout the universe, or is Earth's microbial abundance extraordinary and rare? Every ocean, every handful of soil, every breath you take might be filled with either cosmic commonplace or miraculous rarity, and we genuinely don't know which. The universe might be teeming with microorganisms, every planet with liquid water hosting bacterial ecosystems, life as common as stars. Or Earth might be unique, a singular accident in billions of light years, the only place where chemistry achieved biology, where matter became alive. The answer determines our cosmic significance. If life is common, we're participants in a universal phenomenon, part of a pattern repeated across the galaxy. If life is rare, we're precious, perhaps unique, carrying responsibility for biology's continuation, guardians of the only consciousness the universe has produced in this corner of space and time. We search for biosignatures on exoplanets, for life in Mars' soil, for microorganisms in Europa's ocean. Each negative result might mean we haven't looked carefully enough, or might mean life truly is rare, that Earth's abundance is extraordinary. Each potential positive, if we ever find one, would answer the question definitively, prove that life arose at least twice, suggest it's probable rather than miraculous. Until then, we exist in uncertainty, surrounded by trillions of microorganisms whose cosmic significance remains unknown. They might be utterly ordinary, representatives of universal biology, or they might be miracles rare beyond comprehension, life that happened to emerge here and might exist nowhere else. You're breathing them now, hosting them, depending on them utterly, these ancient invisible things that built the world, that might be common as hydrogen or rare as consciousness itself. We don't know. We can only wonder and search and wait for the universe to tell us whether the microbial abundance we take for granted is everywhere, or only here, in this particular corner of an otherwise sterile cosmos. And so we've travelled through the invisible world, through billions of years and trillions of organisms, through extremes of temperature and pressure and chemistry, through partnerships and warfare and transformations that shape the planet and everything living on it. These microorganisms, too small to see, too numerous to count, too diverse to fully catalogue, are the foundation of biology, the pioneers of evolution, the ancestors of all complexity. They were here first, 3.5 billion years before you, shaping the atmosphere you breathe, establishing the cycles that sustain ecosystems, inventing every essential biochemistry that life depends on. They live within you, around you, beneath your feet and above your head, in clouds and caves and oceans and soil, in places too hot or cold or acidic for anything else, testing the limits of life, expanding those limits, proving again and again that biology is more tenacious, more adaptable, more widespread than we ever imagined. Perhaps they're everywhere in the universe, common as water and carbon. Perhaps they're rare, a fortunate accident that happened here and nowhere else. We don't know yet, but we know they're ancient, resilient, essential, invisible architects that built the visible world, that maintain it still, that will likely persist long after larger, more complex organisms have come and gone. Tonight, as you drift towards sleep, you're not alone. You carry trillions of them, descendants of Earth's first life, partners that help you digest, that protect you, that produce vitamins you need, that constitute half the cells in your body. You're breathing oxygen they produce. You're depending on nitrogen they've fixed. You're part of systems they created and maintain, a temporary participant in microbial processes that predate and will outlast humanity. Rest now, knowing you're surrounded and supported by ancient invisible life, by organisms that have survived everything Earth could throw at them, that adapted to every extreme, that found ways to thrive in conditions that seemed impossible. They're patient. They're persistent. They've been here through ice ages and asteroid impacts and volcanic cataclysms, and they'll be here long after tonight becomes tomorrow, continuing their quiet work of transformation, their endless cycles of living and dying and living again. Good night. Sleep well among your microbial companions, these invisible nations that call you home,